who's going to give his opening speech for us today. Mr. Yudi, are you here?
Hello, good morning, everybody.
Good morning, Mr. Yudi. All right. How are you today, sir?
Oh, fine, very well. Thank you.
All right. To Mr. Yudi, the screen and time is yours.
Okay, thank you. Yeah. Very good morning to all of the participants. Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to greet to all of the participants, especially to the speakers, the professor for second. Yeah, to all of the speakers, yeah, Pak Noir and all speakers. I hope this is not the last meeting, but this is the first meeting, and after this event, we can create for the strengthening the collaboration between the Marine Science Study Program and also the Faculty of Fisheries and Marine Sciences Universitas Pelajaran to create in the future for example, for research, publication, and the things is how to make our ocean better in the future. And then the second one, as we know that the ocean will become the biggest economic growth in the future. And if we look to the mega trend, the global mega trend 2045, there will be a competition in the global community to compete in natural resources. And ocean is one of the biggest potential natural resources in economic and ecology. So that's why this is the important things, I think, to discuss how we can manage this ocean to have the economic and ecologic, and finally for the welfare of the community. And then the third one, I think the Faculty of Fisheries and Marine Sciences Universitas Pelajaran, through the Marine Science Study Program, we have an international class in the next term. That this program is, we want to create the human capital who will be the leader in the future, who can manage the ocean and giving the economic and ecologic to have the world better in the future. So I hope all of participants, especially from the high school, we will ask to join in this program, and we will create this international program as the better class in ocean in Indonesia. We have a collaboration with the institutional education in abroad, for example, in Tiongkok, in Japan, and in Korea. So through this international class, we can send and make the words smaller, as we know. So this is the third things that I want to giving as an opening speech in this morning. So my summary is first, to all of the speakers, I hope this is not the last meeting, but this is the first meeting. And after this event, we can create new program for the strengthening the collaboration between us, among us. And then the second one, let's and enjoy discussion to have a summary how to manage ocean better in the future for giving an economic and ecological for welfare community in the future. And the third one, we will ask to all of participants from high school to join in international class of Marine Science Study Program, and being the human capital who can be a leader in ocean management and be the best class in the world. So I think by saying in the name of God, let's start this webinar, this event, and enjoy to all of participants. Thank you. Billahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
What a wonderful opening speech. Thank you for giving the speech for us today, Mr. Yudi. So we're going to have a photo session with Mr. Yudi. So to all participants, please turn on your camera and put on your biggest smile. There is 8 slided in total, so we're going to take photo 8 times. The command will be on me. Okay, for the first slide, 1, 2, 3. The second slide, 1, 2, 3. The third slide, 1, 2, 3. The fourth slide, 1, 2, 3. The fifth slide, 1, 2, 3. The sixth slide, 1, 2, 3. The seventh slide, 1, 2, 3. The eighth slide, 1, 2, 3. Okay, that's it for the photo session. Once again, thank you, Mr. Yudi, for today. So for the next session,
allow me to leave because I have a graduate ceremony today for alumni from FPIK UNPAD. Thank you.
Okay, thank you, Sir Yudi. Okay, so for the next session, we have Lecture 1 and Lecture 2 that will be shared by Mr. Andersson Mayfield, Ph.D., and Andreas Andersson, Ph.D., with the moderator of Mr. Buntora Pasaribu, Ph.D. Before we hand it over to Mr. Tora, we would like to read his CV first. Okay, so Mr. Tora is an assistant professor, Department of Marine, Pajajaran University. His background education, doctoral degree from PhD in Biotechnology National Chung Sing University, Taiwan, postdoctoral degree, Department of Plant Biology, Rogers University from USA. His expertise is from part in at the genetics and oil bodies in coral, algae, marine plants. Okay, Mr. Tora, are you here?
Yes, I'm here.
Good morning, Mr. Tora. How are you?
Yeah, I'm good. How are you?
I'm good too. So happy to have you here. So I'm going to hand it over to you. And thank you, Mr. Tora, for facilitating our first lecture today. Mr. Tora, the time and screen is yours.
All right. Thank you so much for Regita and our MC today. I think this is the great opportunity for us because we have one of the top marine researchers and his expertise in coral, and he has been a founder of Coral Diagnostic. That I would like to welcome Dr. Andersson Mayfield. Are you there?
I'm here. Hello, everybody.
May I? Oh yeah. All right. It's a long time no see.
Yeah, good to see you.
Yeah. All right. Yeah, I think this is the very interesting topic that Dr. Andersson will deliver. The topic about the coral and his topic about leveraging the power of artificial intelligence to cover resilient reefs in the coral triangle. And before we start Dr. Andersson presentation, that I would like to read his CV briefly. Dr. Andersson is currently chief scientist at the Coral Reef Diagnostic, and then that he obtained his PhD degree in zoology from University of Hawaii, Manoa, and his expertise in genetics, coral, and molecular biology. Andersson, please take away.
All right, let's get my slide deck up here. Are you seeing it okay?
Yep. Nope, that I'm still seeing your face on the screen, so I think.
Oh. Let's see. Sorry, everybody.
Yeah. Don't take your time.
Oh, wait, here we go. I think I got it now. You see the slide now?
Not yet. Okay, now, yes.
Okay, I think there's a little bit of a lag but I think it's going to work. All right. Well,
All right.
thanks everybody. This is no, this is a much better turnout than I expected. My name's Andersson. As Toro said, I've been doing coral reef research for a number of years. We actually overlapped in Taiwan for at least a couple of years. I was in Taiwan for a long time, and my background is coral molecular biology, and I'm going to talk about that today. But I'm going to the way my talk's going to work, I'm going to kind of switch gears halfway through and talk about some of the new stuff I'm doing, which is, it's all getting at the same issue of how do we predict what the future will look like for coral reefs? Which reefs will survive, which won't, which species will be around? It's all getting at kind of the same topics and questions, but in my past life, I was doing it exclusively with physiological and molecular tools. And recently I've been using more of computer science, big data, analytics, artificial intelligence kind of stuff. So I hope, at least half the talk might be something that you're interested in. Maybe you'll be interested in the molecular part, maybe you'll be interested in the modeling part. So I don't think I'm going to go into too much detail because I think even the youngest of you are going to be pretty familiar with these problems facing coral reefs, notably climate change. What I was doing in the past was a little bit smaller in scope. I wasn't looking at the whole reef. I was really just looking at the individual corals. Can I tell you what's going to happen to this coral? Is it going to live or is it going to die? What about this coral over here? Is it healthy or is it not healthy? That's kind of what I was doing a long time ago, and I'm still working on this. But now I'm looking at a bigger picture. So not just coral by coral, but the entire reef. Is this reef healthy or is it stressed? Is this reef resilient and can handle the change in its environment, or is it going to perish in the coming decades? So, for the longest time, we were going out there and we were thinking, all right, reefs near the big cities are probably going to die first because, there's heavy human impact, there's seawater pollution, there's climate change. So let's go out there and go to all the reefs that are in the middle of nowhere, far away from people. Those are going to be the quote unquote pristine reefs that we should focus our efforts on. Unfortunately, it ended up being kind of the opposite. We went out there all over the Indo-Pacific region as a part of a series of research cruises. I'm going to show you some of the data later. And these reefs that have never experienced any stressors before, they're actually not very well prepared. So it's kind of sad because I was really hoping that reefs would be okay as long as they're really far away from people. But the sad thing is climate change, is it knows no, international boundaries. Some of these atolls that are hundreds of kilometers from cities, they're doing just as poorly, if not more poorly. But we know that there's strong corals out there. If you go diving or snorkeling, you see this situation all the time. These are not even just the same species. This is the same clone. Why did the half on the right become bleached and white and sick when the temperature got too warm? And the exact same, so its genetic twin, seemingly similar environment, why is it doing better on the left? So this is kind of one of the questions that drove my research in before. So what's the physiological explanation? And is that something we can predict? When these corals are both healthy, can I tell you which one's going to be the white one and which one's going to be the healthy brown one? So later, I'm going to show you an example. I'm going to show you two different examples of how I've been doing this. And this is just a lot of these pictures. Actually, I'm a really avid scuba diver, and my favorite place for diving is Indonesia. I do, this is the country that has the largest coral reef area, has the highest biodiversity, pretty much every metric of grandeur for coral reefs. Indonesia is going to be number one, and I think probably some Indonesians don't realize that, and certainly a lot of Westerners and Americans don't know this. And not a day goes by where I'm not showing people pictures of places I went diving in Indonesia, or talking about how much I love Coral Triangle, Philippines as well. So I think, you know, and this is the area that I get the most excited about, because yes, we have all these same problems, rising seawater temperature, pollution. It's not to say that I'm not worried about the coral reefs in Indonesia, but I see chances, I see opportunities, I see reasons to be hopeful. You know, some of the reefs that I work on here, and I'm in Florida now, so the Caribbean, they're good for doing research and publishing papers, but they're beyond saving. You know, doesn't mean we should stop studying them, but they don't really give me any hope for the future. Coral Triangle I gives me a little bit more optimism. I think there's not as much disease issues as we're encountering here in the Caribbean. So I think for me, you know, talking to primarily an Indonesian audience, I think, you know, there's a lot of reasons to be proud and to really want to cherish these ecosystems. I mean, you're going to see almost every beautiful picture of healthy reefs that I'm going to show in this talk, except it's going to be from Indonesia, or maybe when I talk about the Solomon Islands, I'll show Solomon Islands photos. So I hope if you're not big into diving and snorkeling yet, and you're just getting started, that some of these pictures will inspire you. Okay, so getting back to the science. So the first kind of 20 years of my career, I did what I facetiously call writing coral reef obituaries. I baked corals at high temperatures. I cranked up the PCO2. I slowly killed corals to see what would happen to them. And I don't want to belittle this, because this was important research and we needed to know how these corals would respond. But really, I was like a coral undertaker. I was just documenting death, and it was good for my career, but it was kind of depressing. And by the time I wrote these articles, the corals were already dead. So like I said, we needed this kind of research to be done, but it wasn't always the most gratifying as a conservationist. So what I've been trying to do in the last few years is, rather than an undertaker who's burying dead coral, I'm a veterinarian. So I'm treating these corals that are still alive like my patients. I want to know what I can do to help them get better. And analogously, instead of writing coral reef obituaries, where I'm just telling you how many corals are dead, I'm writing an epidemiological report, trying to show you the trajectory that that reef is on. Is it on a downward spiral? Is it on up and up? And this is getting at these things like, can we find resilient corals in places where we've never done any research? There's no way we're going to have time to survey all the world's coral reefs before a lot of them die. We need a faster way to do this. And for those reefs that aren't strong, is there something we can do to rescue the corals or make them more resilient? So this is a lot of the stuff I've been doing that's really kind of taken a data science approach. So I don't want to, you know, make this seem like I'm the first person that ever tried to look at the future of coral reefs. No, NOAA's been doing this for 20 years. They look at temperature trends, because even though corals live in the tropics, their thermotolerance is relatively narrow, and they don't like water much above 1 degree Celsius of kind of their normal summer mean. So NOAA took that information, combined it with their satellite temperature data, and said, look, we can predict bleaching. We don't need all that fancy stuff you were talking about. Let's just look at temperature trends and see if we can predict timing of bleaching. And it's a really nice website. They've got real-time updates. They basically show you values. It's called the degree heating week. It's basically how long your reef has been at high temperatures. And once you start to get in the degree heating week levels of 4, 5, 6, 7, 8, their model is going to predict that your reef is going to bleach. And in a lot of parts of the world, this is a great model. You don't need to, you could sit at your computer and not even get wet or do any lab work and just say, hey, you know, this model's telling me my reef's going to bleach. Let's go out there and check it out. You know, they make it easy for you. It's very egalitarian, but of course it's an oversimplification. And I mean, and that's what models are about. They're trying to distill something complex in, you know, based on something simple, but which I respect, but I think we can do better. We can make it a little bit more complex, but also get a better predictive outcome. So these models, they don't care what corals are there. They don't care if your reef is 100% weedy acroporids or 100% massive corals that do better. They don't factor that in. They're just looking at temperature. They're also, in the few instances where you see something beyond temperature in a predictive model, they typically only focus on quantity, coral cover, coral cover, coral cover. You need to measure coral cover. And I definitely don't want to leave you, here saying, oh, this Andersson guy said we shouldn't do benthic surveys anymore. No, that's not what I want to say. But I do have an issue with basing the resilience of a reef solely on the abundance of coral. And this is a little bit of a blasphemous issue, because normally your brain as a tourist and as a scientist, you go to a reef like this is in Maluku, and you say, wow, it's 100% coral cover almost. This is a healthy reef. Okay, healthy might be the right word. It might be highly valuable for tourists, but this is not a resilient reef. It's a high coral cover reef. It has high ecosystem services, but this reef may not be around in the future. I'll show you, this is actually not a great example because there are some resilient corals in this picture. I'll show some other examples later of a reef that's got super high coral abundance, very low coral resilience. But basically, I think, okay, maybe 20 years ago, it was forgivable to say, hey, we're doing the best we can. Let's just look at temperature. Now, there's a lot of people doing coral reef research. We have big data sets now. We can start doing more than just looking at temperature and coral cover. We can actually start to consider the health of the corals themselves. Oh, so this is a better example. So this is in Papua and Raja Ampat. Every single coral, there's only 3 or 4 species in this picture. It's beautiful. This is one of the most famous coral reefs in the world. It's called Melissa's Garden. This reef is, again, very high abundance of coral. The invertebrate biodiversity is going to be really high. This reef would bleach very quickly if you know the species that are living here. So this is a reef with high ecosystem productivity, high tourist value, but I don't know if it's going to be there in 20 years. So I think people need to kind of move beyond this idea of more coral equals better. If you think about it, we don't think of cities that way. I don't say, oh, the people in Los Angeles is a bigger city than Miami. That means the people in Los Angeles must be healthier than the people in Miami. Maybe the people in the big city have access to better healthcare, better food, but maybe the big cities also have more traffic and more crime. So I think it's strange that for people, we don't equate city size and health of the citizens, but that's what we do in the ocean. We say, oh, more coral, that's a healthier reef. And I think this is like the kind of argument where you don't win any friends making this kind of argument, because it's like, it makes for more work and it's kind of blasphemous. But I just want to say that I think, we need to know what corals are there and the abundance. We need to know the temperature. So it's not to say that none of those things are important, but I think what we can do is try to integrate these things and see if the predictions we get are telling us the same thing. So I've kind of grouped it as this. I've got the abiotic data. This is mainly seawater quality, the temperature, things we know are important for coral health. The ecological factors, this is basically our survey data, coral cover, coral diversity, fish biomass, algal biomass, all these things we know are important for reef health. And also the physiological data from the corals themselves, which used to be too hard to acquire or too expensive. So we kind of discounted that part because we just didn't have enough data, but now we do for a lot of places. So I'm going to give you an example of what you would do if you only had the coral health data. Maybe you've got some seawater data, you've got some survey data, but you're trying to make predictions about the health of a coral looking only at the corals themselves. And I call this the veterinarian's approach. This is using the same approach your doctor would use. They draw blood from you, they do some tests in the lab, which is what I was doing when Turo and I were in Taiwan, where I was basically developing lab tests to look at coral stress. And then I can give them a grade, for instance. Maybe they're an A, or they're a high score, so they're more resistant. Or maybe they're an F, This is using the US grading system, sorry. And they're going to be likely to bleach. It doesn't matter. At the end, I want to be able to just give a single number that will tell you about the health of this coral. And, you know, I'm going to make this seem like it was really simple, but this took like 20 years of lab work. We needed to develop the methods. We needed to figure out what are the best things to measure in the coral. You know, the corals don't have blood pressure. There's a lot of things we measure in humans that don't exist in coral, or they're not important for knowing the health. So Toro and I and the people we worked with, this is a lot of what we were doing, looking at, you know, what lipids are important in corals, what proteins are involved in the stress response. This was stuff that we didn't know 10 years ago. So we had to go do kind of this fundamental research, but now it's paying off because we can start to do more powerful stuff. So like I mentioned before, my background's in molecular biology. And the reason I think molecular biology could be useful in coral health monitoring is things change at the molecular level before they start to show up at the physiological level. Your blood pressure, for instance, might be really high. Maybe you still feel healthy. You didn't have a heart attack, but your blood pressure might start to become high weeks, months, even years before you have a heart attack. So you go to the doctor and they can know something about your health by taking your cholesterol or blood sugar before you get sick. Then maybe you can change your diet. You can take medicine. You could do something to make your personal health better. So for corals, we usually don't do this. We wait till the coral's dead, and then we say, oh, that coral was stressed. Obviously, that would not be acceptable for people. If you waited till you had a heart attack or died, and then your doctor told your family, oh, yeah, he had high blood pressure. You want to know that before you get sick. So that's why I think molecular biology is important. I spent, it's 9 o'clock here in the US. I spent 15 hours today training students in what we call multiomics. Don't worry, we're not going to go through it. But because corals have dinoflagellates that live inside of their cells, you need to modify a lot of the protocols to get good quality DNA, RNA, protein, stuff like this. So that, again, this is a lot of what Toro and I were doing in Taiwan, developing these protocols, figuring out what to measure, a lot of lab work. And anybody who's interested in lab work and molecular stuff, definitely email me and I'll flood you with all kinds of information. But I'm going to try to keep the molecular part light today. So what we would do here in Florida is, the good thing about adult corals is they don't move. So I know where these corals are. They're my patients. I can go out there and find them. I can take tiny little samples. So I don't need to kill the coral, just tiny little polyps, take them back to the lab, do my laboratory analyses, and then I can go back in time. So I know when the coral bleached, so I can go weeks, months, years before that coral bleached and start to look into its cells and see when things started to go wrong. When did that coral start to become stressed? And unfortunately, I don't know any good way to portray this, but this is where the machine learning came in. So this is actually, this example is what's called a neural network. It's a type of artificial intelligence. It's actually been around for 30 or 40 years, but it's kind of become trendier lately because it's being used so much in like image analysis and advertising. You can use this. It's really just a sophisticated modeling tool. And it's looking through my data from my corals and trying to make predictions about which corals are going to live and which corals are going to die based on these little samples I took and looking at, in this example, it was proteins. But you could do the same approach with gene expression, lipids, anything you want to measure that could be useful for coral health. You could build these artificial intelligence models. And it's all I've been doing for years. And, it's obviously more advanced than just hitting a button, but I think you'd actually be surprised how quickly you could use something like this to better understand your target organism or your target ecosystem. And then at the end of the day, I can have that artificial intelligence that I've been training. The idea basically in a nutshell is you give the artificial intelligence a lot of data and you hide data from it. And you say, all right, don't look at this data. Just look at this data and make a prediction about these over here. And if the artificial intelligence is correct, then obviously that's good. If it's incorrect, it can actually try to learn and improve to do better the next time. So that's why they call it machine learning. So basically, I've been using machine learning or artificial intelligence to basically make coral health checkups in the Florida Keys, giving them grades. You know, the A, this is just hypothetical data, but in this example, you know, the A, that's where we're going to find the best corals, the most resilient. The F, that's where we're going to find the wimpiest. And you know, if I told a manager this, they might look at that F and say, you know what, that reef's already beyond saving. You know, there's nothing I can do. A manager might actually be most interested in the ones in the middle. Those are the like the A plus, those are fine. All right, we don't worry about those. But the C, maybe that's the one where you want to focus your effort, because that one, maybe it still has a chance if you did something to help the corals. This is the big elephant in the room, though. I was working at NOAA before. NOAA has a lot of money. That project, all these last few slides, was basically a 3-year project with NOAA. It almost cost $250,000 US. It was only for one species in one place. Think about that. It's 500-600 coral species in Indonesia, which is a much larger area. By the time, even if you had the money to do this for every species, by the time you finished it, you know, it would be 300 years in the future. It's too slow and it's too expensive, frankly. Even in the US, it's too slow and too expensive. We need a faster way than doing this exclusively like diagnostic veterinarians approach. So that's why I was saying, it's not that I completely abandoned molecular research. I value those experiences, but I'm not sure it's necessarily the best way forward because it's slow and expensive, and that bothers me. I want to have a predictive model that's faster and doesn't require weeks and 80 hours a week of lab work, because I'm getting old and I can't do this anymore. So I need a faster way. So usually what we do when we find resilient corals is we find them by accident. We go diving on a reef after a bleaching event and we say, oh, look, that coral over there survived the bleaching event. We need to study it. That's great, but you need a way to try to find this before the bleaching event. Or you take the coral into the laboratory, you do an experiment, then you find out which ones are the strong ones, which ones are the weak ones. Again, you're not going to be able to go to every reef in Indonesia, take a couple of corals, bring them into the lab, and look at their thermotolerance. You would need too many people, too much money, and too much time. You need a faster way to find out where are the strongest corals in the coral triangle in this example. So what I've been doing for this is, if you remember before, I only looked at the red circle, the physiological data. But at the beginning of the talk, I kind of mentioned, hey, what if we combine all these different types of data? The seawater quality, what's living on the reef, the stuff we know about the coral. This is what epidemiologists do. When they're trying to track trends in COVID, for instance, they're looking at all different kinds of data to try to see which city is going to have the highest infection rate, what month is going to have the highest infection. They've been doing this for years with human health data. So I've been trying to adapt it to coral health data. And I really wish, and I really hope that I can do something similar in Indonesia in the future. I do have some really good collaborators there in Ambon, and we just submitted a proposal to do a little bit like what I'm going to talk to you about in a minute. But today I'm going to be talking about a different part of the Coral Triangle, which is the Solomon Islands. I went there as part of this global reef expedition a few years ago. It was the largest ever coral reef survey. So that's why I'm saying we didn't used to have all this data. Now we do. So we can start to do a lot of cool stuff with data science and predictive modeling. So we did all the stuff you might be used to. Mapping the coral reefs with satellites and by boat. We did our diver surveys to see what's living on the reef. We looked at the fish. We took the seawater quality. And then I was taking my tiny little samples and doing my molecular analyses. We went all over the country. It's a really beautiful place. You know, it's got Some of the reefs are almost as nice as ones you'd see in Papua and Maluku, and even Sulawesi and places like this. So what I did is I didn't really go into the details, but in the Keys, I mainly looked at proteins. In the Solomon Islands, I looked at this benchmark I created called the Coral Health Index. And don't worry, again, this is kind of complex by design. It's basically a ton of different biomarkers and different things I measured in the coral to give me an idea of the health. So the outside wedges that you see, the average is what you see in the middle, that average score. And the Coral Health Index arbitrarily goes from 0 to 5. 0 means you're about to die. 5 means you're probably going to live forever, because if you don't know, corals technically are immortal. They don't age. The only reason they die is usually when the environment changes. So we went and took tons of samples and looked at the Coral Health Index scores across the Solomon Islands. And this is not what I want to leave you with. This is not that interesting. The only thing interesting about this figure is the fact that the higher scores are in the east. And that's where you basically see fewer people. So even though at the beginning of the talk, I say, there's not really any pristine reefs left, I do agree with that. But in the case of the Solomon Islands, this trend does kind of play out. So most people in the Solomon Islands live in the west, and the reefs are doing a little bit worse there. It's not really hugely different. 2.5 versus 2.5 to 3. Anyway, this is not the take-home message. This is what I used to do. I would just do like a baseline health assessment, but I want to do more now. So instead of just telling you the Coral Health Index score, this might be okay for a publication, but what I want to do is try to predict all these places in the middle, maybe, for instance, that we didn't go to. What's the coral health there? Because Solomon Islands is a tiny country compared to Indonesia, but there's no way, even though we spent 2 months here, there's no way we saw every reef. So how do we fill in gaps? How do we predict the health of the coral we didn't survey? That's what I think will be cool, because it would take years to survey all the reefs in the Solomon Islands, decades to survey all the reefs in Indonesia. So that's not going to be the most productive way. You want to immediately target those resilient reefs. If you only have 2 weeks of field time, you want to go directly to that super reef, or maybe you want to go to the weakest reef. You want to know which reef to go to before you start your field trip. So again, I went back to the machine learning. I built all these neural networks that they don't actually require that much computing power, but they're really complex to interpret. But I basically used all those data we collected to predict where we'll find the resilient corals and in the Solomon Islands. And this is kind of what some of the output looks like. So you see things like latitude, longitude. So this is telling you, as you go from -11 to -8 latitude, the Coral Health Index decreases. So in the Solomon Islands, the highest coral resilience you will predict is around -12. Another example, all right, lots of different types of reef. Which reef will you predict has the most resilient corals in the Solomon Islands? Fringing reef, maybe not by a lot, but the model's telling you you should go to a fringing reef. This is interesting. And this is getting back to what I was talking about. Barren substrate. That means the part of the reef that has no corals. It tells you to go to the very highest values. So what that means is the most resilient corals in the Solomon Islands are the ones that have the fewest number of corals. And I show that another way in a minute. Actually, I'll just show it now. So that's the data. So when I was saying earlier, there's no correlation between coral cover and coral resilience, you could see it's basically 0. So another example, this reef is maybe in Bunaken. It's beautiful. It's high coral cover. Tourists will love to see this. May be resilient, but it may not be. I wouldn't judge its resilience just based on the coral cover. So you can play around with these things. It's called a desirability analysis. You can basically make an app. That's what I'm doing now. I'm putting these apps up on my website. So then you could enter in the data for your reef, and it will make a prediction about what the resilience will be like at those reefs. And I think that could be really useful for people. And I want to do something kind of like what NOAA did is, you know, I have a coral health map, but instead of just looking at temperature, I'm looking at all these different things. And, you know, in a lot of cases, after all this work, my model might say the exact same thing as this 20-year-old one. But I bet you there'll be other instances where our models disagree. Maybe there'll be some instances where the NOAA model says, hey, this reef's going to bleach, but mine tells you it won't, or vice versa. So it's not to say that I'm trying to supplant or replace this. I think they're both useful, but I think there'll be situations where maybe you need a higher level of spatial resolution, for instance. This is not making predictions about corals or reefs. It's making predictions over kilometers, tens of kilometers. So my efforts are a little bit, you know, I want to go global in scale, but right now I'm kind of working at the island, the archipelago level. So I'm not at the level that NOAA's at yet, but the idea would be to grow it into something like that. Because these models are going to tell you where your most resilient corals are and where the weakest ones are. And this is going to let you make predictions about the future. It's going to let you know which reefs will still be around and which won't, which ones might have the best genotypes for restoration. This is going to be a great tool for bioprospecting or determining the optimal environment for a coral nursery. It's going to help be useful for mitigation. And it could also, I'm not going to have time to talk about it today, but could also help dictate, all right, given what we know about this reef, which conservation strategy has the highest chance of success? Is it coral nursery? Is it MPA? Is it antibiotic treatment? These are the kinds of questions and kinds of conservation strategies you can really tailor using these kind of machine learning approaches. So I have this whole how to save a coral flow chart where you go from the top is the best case scenario. Best case scenario is you stop climate change. Obviously, that's might be unrealistic at this point. As you progress through this flow chart, it becomes more and more dire and more kind of late stage measures. So where Toro and I worked in Taiwan is the world's premier facility for coral cryopreservation. I don't want to have to cryopreserve coral. That's sad. To me, that represents the end game. All right, we can't do anything. Let's just put the corals under the ice and hope that an alien race can thaw them out in 1 million years. But that's the kind of things we're planning. We have to have a backup plan in case all this other stuff fails. This was just to take you through some stuff on my website where you can get more information, but don't worry, we're not going to go through with that. And this is just a little bit more about where Toro and I were working in Taiwan. Even though I don't work there anymore, it's a really great place. Especially Taiwan has this Southeast Asia initiative now, but they really want to have a better relationship with Indonesia, Philippines, students going back and forth, training. So I think if anything in this talk interests you, by all means, keep it local. But if there's something, maybe cryopreservation or coral molecular biology or some tool that's maybe you don't find in your local university, start maybe looking into Taiwan, and there might be some opportunity there for you, all different coral husbandry, cryopreservation, genetic modification, other stuff I haven't talked about, all these really cool cutting edge coral stuff. I really recommend Southern Taiwan and Taiwan in general if these things pique your interest. And it's a lot of the same corals. There's other good reasons to go there. People are nice, food's great. Okay, so I know that was kind of a whirlwind, a lot of stuff covered, but if you don't remember anything else, just remember that I had my one approach where I used a molecular method plus an AI to try to make predictions about my patients. That was what I call the veterinarian's approach. That was the approach that's too slow and too expensive. So I still help people out with that. I still do a lot of molecular work, but I'm starting to worry that this approach is just not going to cut it. We're running out of time. So now I'm doing this epidemiologist approach where I'm really trying to look more globally and make predictions about coral resilience that I think are on a time scale that's more commensurate with the rate at which reefs are declining. And now what I want to do is use kind of this data science, machine learning pipeline to try to optimize coral rescue. This is fun. Last time I was in Indonesia, I worked with some people to write an article. This is a different topic. This is kind of one of the topics I was talking about earlier. I touched on that there's no more pristine reefs left. People will fight me on this because it helps them get grant money, but it's true. But yeah, I encourage you to read this. It's a kind of a fun article. It's not too dense. Yep. And
Right.
that's pretty much all I wanted to talk about. And this is also from Raja Ampat, by the way. And yeah, I'll be happy to take any questions, and thank you all for tuning in.
Thank you so much, Andersson. I think it's a great talk, and it is a lot of the information. It's really surprised me for all the slide and then the development that you have been working so far in many years, right? Yeah. So can we give applause first to the Dr. Andersson for his great talk, and now that we will do the Q&A, so that we only have a limit. We only have a limit time right now, so we will have the two section of the Q&A. So the first section is only for the two questions for the two persons, and then the next the second section is limited only to the two persons. Yep. Yeah. Is there any question for Dr. Andersson? All right. I'm gonna ask the question first, Andersson, right? I think, and so your target, I think that what your purpose is it? I mean, is it possible to make all the data that you have been collected so far in many years to be the personalized coral health in the future? Because as we know, we have the personalized medicine, right? And then that we can know from the beginning until to the end of the human, and then they know that which is the cancer, what's the disease that the one person have. And now that is it possible that the AI can predict and then they can build one of the personalized coral health? Yep.
That's a really, really good question. And that's the funny thing is another NMMBA. So where Toro and I worked was NMMBA, the Taiwan Aquarium. There's a guy we both know there, Dr. Joshing Lin. He's the guy doing the cryopreservation. So Dr. Lin and I, a few years ago, wrote kind of a fun article on this topic. We called it the Coral Hospital. Some of the slides I showed are actually from that article. And that was one of the things we proposed, was basically, we didn't call it personalized medicine, but the idea was, okay, what about things like probiotics? You know, maybe exercise doesn't make sense for coral, but there's some things we do. Medicine is the most logical one. And a lot of the stuff we proposed was really kind of just, we believed in it, but we didn't know people would actually try it. But after we wrote the article, people started doing this, like probiotics, giving corals like better nourishment and stuff. And I got really excited about it, because one thing we were trying to do is, for instance, this is kind of, it sounds funny, but I think it's cool. What happens if you make a coral really fat? You give them so much food that you might predict, hey, if that coral has a lot of lipids, a lot of energy, maybe, you know, being really fat is not good for us. But what if you made a coral really super chubby, and then there's a stress event? That might actually be good for the coral, because then it has all this extra lipid energy to help it stay, you know, keep it energetic, its metabolism going, even though it's having problems with the high temperature. So if you know, like the French food, they have a foie gras. They force feed the duck to make their livers or their spleens become huge. It's actually kind of cruel, but we were thinking about doing this with coral. Can we force feed the coral to basically make them super well nourished, and then maybe they will be stronger and can better handle the stress? And truth be told, we tried it and it was interesting. It didn't work that beautifully, but I haven't given up on it. I think there's stuff you can do to treat a coral with different food, different medicine, different probiotics to help it be stronger. I think this is a super interesting topic. And if yeah, I hope some of you listening, if this piques your interest, definitely reach out to me or yeah, I think this is something people should be working on.
Yeah, I think it is, right? And before that, I have a, is there any question before that I ask the second questions? Yeah, because that I want to ask more technically, Andersson, about that your AI. I think that why you are look at the physiological rather than the combined the three factors that you are mentioned in your previous slide. Why you are focusing is only in the physical factor in this.
Yeah, it's funny because I almost removed that part from my talk because I was like, you know what? I know at the end of the talk, I'm going to basically tell everybody, yes, that method is very sexy, but it's too expensive. But I wanted to, I don't want to leave you feeling that molecular biology no longer has a role in coral conservation. I don't want that to be the take-home message. I want the message more to be that there's different ways you could go about looking at coral health. Maybe there's no one right answer. I think that's really what I wanted to do. So maybe in places like Japan, Taiwan, parts of the US, maybe it still makes sense to do this kind of molecular approach, because then you can get much higher prediction. So you could still make a good case for doing the physiological molecular approach, maybe in certain instances. But when you're looking at huge areas like the Great Barrier Reef, there's a lot of people that study it, but even then, and they have a lot of money, even then, it doesn't make sense to go sample 20,000 corals on the Great Barrier Reef. Like there's better ways to get at the health of those corals without invasive sampling. So I think it's not to say that molecular biology no longer has a place in this field. It has kind of more to do with the scale. So I want to make bigger picture predictions now. 10 years ago, I really just cared about the corals in a small area. And then maybe the molecular approach makes more sense. So I think it kind of depends. If you're, for instance, if you have a coral nursery and you only care about the corals in your nursery, the molecular approach is still actually really good because you only need to worry about your coral patients. So you can get really high resolution data, especially if you can afford it. So I think, I don't want to say one approach is better than the other. I just want to say, if you want to look at an area the size of the coral triangle, then maybe molecular approaches will just be too slow and expensive. But if you've got a coral nursery or a hotel, if you've got a house reef, there's plenty of instances where the molecular approach, I think, still you might want to explore. So I'm glad you asked that, and I could, leave with that and not totally saying the first 20 years of my career was like a waste of time. So.
All right, Andersson, thank you so much. Got it. Yep. Think. Is there any question for Dr. Andersson from participants? Yeah. That we have. We are now in the section in the second section, so that you feel free to ask question. I think from participants. Yeah, we can. We can take up like one or two persons. I think this is the great talk, and this is the very this is the excited topic at the and the new milestone research from Dr. Andersson, because that he started to think about the bigger picture of the corals and to protect the corals for the future.
It can be hard though, because I think it's not common for people to completely switch their research direction.
Absolutely.
But I don't regret it, and I think I will say honestly, like if feel free to ask a question. Otherwise, I'm just going to keep blabbing. But I will say that is, if I was to leave you with some advice, it's because I see a lot of young people here, or mostly young people here, which is great. Is doesn't even have to be AI, but most people listening here are already better with computers and that kind of stuff than me. Like, you're going to be able to do so much more stuff with data than I could, that would take me 3 years. Most of you might be able to pick that up in a couple of months. So I think statistics and these kind of courses were historically not very popular. They're like the ones biologists dread. You want to see the pretty coral and stuff. You're like, numbers, math. But man, if you've got strong training, and analytics and computer stuff, you're going to be able to do so much cool conservation work.
Absolutely. I think especially that we are in the 4.0 era. I think the young generation, I think they are using the computer and then they can do the basic coding compare with our previous era, I think, because now the computer, I think, is really important for the young generation. I think that Andersson, I think that you are opening the window or door for the young generation, especially a lot of the young generation here for the future, because we do not know. I think they're gonna take the all this research in the future to save our ocean, right? Okay. All right. Is there any questions? All right. All right. I think I think they're shy, and I think
or if you don't want to ask, you can ask Toro, and he'll ask it on your behalf. or he can translate for you if you don't want to ask in English.
Then I'm gonna contact you later after this webinar. I think it is for me, especially because we have been working many in Taiwan, and then I think there's one question. Yeah. So the checkboard. Operator, can you please? Could you please open the chat box? Yeah, there's one question.
I haven't been looking in the chat box. Let's look.
And there is one question from one participants. Where is it? The thing?
Yeah, I saw the chat box for a second, then it disappeared.
Oh yeah, maybe Bu Qurnia. Yeah, I think you have a question.
Okay, thank you. This is the question from Trianisa. I will read it. Indonesia has abundant marine potential, but Indonesia has not been able to take full advantage of its potential. What steps should we do, and what the opportunity and challenges? Thank you, Andersson.
Ooh, that's a hard question. Yeah, I mean, it's frustrating for me. One thing I saw that was really cool, and I got to try to think how to say this diplomatically, is this has really nothing to do with research. So Raja Ampat is the highest marine biodiversity in the world. So it's probably some of the most beautiful coral reefs. I mean, I'm sure the data support this, but I'm just talking about purely from my personal subjective opinion. And the reason I mentioned Raja Ampat in particular is what's really cool there is, it's so far from Jakarta. It's not a wealthy area. Like Sarong has some oil money coming in, but they don't get a lot of government support to protect these reefs, which on the one hand is scary because you're like, wow, this is the world's most spectacular reefs. And, it's so physically far, geographically far from the capital. It feels like this forgotten backwater. But the local community and the tourist industry basically came together to kind of police it themselves. And I don't really know all the ins and outs, but I think what happens is the expensive diving liveaboards will pay people to basically not fish or to maybe even do something proactive for the reef. So they're like, they kind of took the power into their own hands, but it was in a good way. And there's places in the Philippines that did this too. They had beautiful reefs and they had issues with overfishing, and the government couldn't stop the poachers, but the diving industry had a lot of money and they would pay the poachers to either work for them or to not fish. There were ways where people were doing stuff at the community scale, even without the help of the government, and they were actually being incredibly successful. So on the one hand, it was frustrating that there wasn't more government support. But on the other hand, it was like kind of heartwarming that they were still able to do a lot. And I was like, wow, they were able to do that much just with the community and the tourism industry. And, that's easier to address things like overfishing. Climate change is going to be much tougher to tackle. But, I think where you see the best success stories are at like these local scale things. Like slowing climate change is not something you can do at a local scale, but you can maybe try to ensure the seawater quality is better. Maybe you could try to reduce agriculture runoff. These things are still difficult, but there's something that a community might actually have a chance to do. And you might, you're going to have to get the government on your side at some point, but I don't think, there's ways you can start making stuff happen without waiting around. It's hard. So I think the only way I can maintain optimism is to just look and see these success stories. You see dive shops going out there, like things Toro was asking about, like coral personalized health. And there's some Caribbean islands where the dive industry, because they're making a lot of money from tourists, they'll use a little bit of that tourist money to go out there and try to help the corals, like culture them, make a nursery. So they're kind of leveraging the tourism money to make small changes at the local scale. So I don't want to say that's the best solution, but that could be a good, like, short-term in a pinch thing, especially these places like some of them are already parks like Bunaken. So a lot of the famous reefs in Indonesia probably are already, they have something like this going on already. I'm kind of more worried about all these other islands that aren't famous yet. Like what's, they don't have tourism money coming in. The people are probably just fishing. Like those I'm a little bit more worried about because they may not have the luxury of foreign money coming in. So yeah, it's easy for me to say as like a white guy in the US, oh, you shouldn't fish there. Oh, you should have better water treatment. You shouldn't have your sewage on the reef. Yeah, that's easy for me to say, but these people are just trying to feed their families. They're not bad people. They don't hate the coral. It's just, so it's hard for us to go in there and tell them not to fish. So yeah, it's hard to make these suggestions to people and not sound condescending. And, I don't know. I think for every success story, there's something that frustrates me. But I think, Indonesia has the world's most spectacular coral reefs. And that, for that reason alone, it should be on every single person's priority, even if you're a businessman and Makassar or something, like, and you never go in the ocean. It still should be like something every Indonesian should know, that their country has the best reefs. Like this should just be common knowledge. And I wonder if there's some kind of a campaign to where there's like a national pride in this. Maybe that would help things. you know, some small countries have done this. They, like Maldives, they're famous for ocean and reefs. They've branded it. People come there for that. So maybe, you know, there could be some kind of like a branding or campaign to kind of really promote this, not just for tourism, but just because it's the right thing to do. So I think the beauty of Indonesia's coral reef should somehow be the selling point. So social media, now there's all different ways to get people excited about this. So I think the young generation are probably going to have some good ideas, like how to make coral reefs more, you know, in the public's mind in Indonesia.
All right, Anderson, thank you so much. I think that we are running out the time, and thank you so much. And
No problem.
can you give applause to Dr. Anderson? Okay. Yeah. All right. Can we, before we go to the next session, I think, can we take a picture, Anderson?
Sure.
Participants? Yep. For the operator, please. Yep. 1, 2, 3. All right. Next. 1, 2, 3. And third. 1, 2, 3. All right. I think that we have many participants. Thank you so much to hear your talk. It's over the 300 per participant. I think
that's great. I thought it was going to be 20 or 30 people. So this is great.
No, it's all right. One more time. I think that we have one more time. Yeah. 1, 2, 3. All right. Yeah, we are done. Thank you so much.
Yeah, no problem.
Yep. And I think we want to show you something because this the appreciation from our institution. I think the operator, can you share your screen? Share the screen, please. Yep. Yes. Yeah. Oh. Yeah. Yeah. Thank you so much. And this is the certificate of the speaker, but for giving the talk for today, Dr. Andersson. I think that we're gonna keep contact, and that we're gonna hope to see you in our next event. Thank you so much for your time. I think.
Yeah. Thanks everybody, and feel free to reach out if you got any questions.
Yeah.
Nice talking to you.
All right. It's nice to meet you too. Okay. If you want to stay, that you can stay. If you want to leave the Zoom meeting, I think that's fine. Yep. We're gonna go to the next session. I think it's one of the alumni from University of Hawaii too. Is Prof. Andersson. Yep. Thank you so much, Andersson. Yep. Yep. Yeah, Dr. Andersson, I think, are you there?
I'm here.
Yeah, Prof. Anders. Yep. Yep. I cannot. Yep. Okay. How are you?
I'm fine, thank you. How are you?
Fine. I'm fine. I'm great. Thank you so much. I think actually this morning I contacted you and I really did not know that actually the daylight saving is starting in the US right now. And yeah, thank you so much for reminding me. All right. And thank you for waiting, because that we have several questions from our previous speakers. Yep. And thank you for your time. I greatly appreciate that. And today that we have a professor, the associate professor Andreas Andersson, that he gonna give a talk. The title is Can seaweed farming mitigate ocean acidification on coral reefs? So before Dr. Andreas Andersson give a talk and start his presentation, I would like to read his CV briefly. Dr. Andreas Andersson is currently an associate professor in University California, San Diego, and also he's affiliated with the Scripps institution. And he obtained his Ph.D. in chemical oceanography from School of the Oceans and Earth Science and Technology at the University of Hawaii. And his expertise in biogeochemical, ocean acidification, and ocean carbon and biochemistry. Dr. Andersson, yeah, please take away.
Okay, thank you so much. I'm gonna try to share my screen. Does that work? Can you see that?
Yep, it's great. Yeah, it's clear.
Okay. Let's see. Okay, well, thank you so much for that introduction, and thank you for giving me the opportunity to talk about some of my research. And the title of my talk is Can Seaweed Farming Mitigate Ocean Acidification on Coral Reefs? And this is part of a project, we call it the OAME project. It's Ocean Acidification Impacts on Marine Ecosystems. And this is funded by the Belmont Forum and the National Science Foundation. And it's a collaboration between researchers in Japan and also in France. And I'm sorry to disappoint you. I'm not going to give you any answers in my talk today. I'm just going to tell you what we're doing and give you a little bit insight to the questions we're trying to address and the methods we're using to address them. And what you see here, this picture is from Andersson in Okinawa and is taken over one of the seaweed farms there. And in the background, you can see the different hotels that are located in the same place. I would like to acknowledge some of my students and former postdocs that have been working with me as well, that are listed here. Before I talk about specifically about this project, let me talk a little bit about what we do in my lab. So one of the underlying motivations for the research we are doing is really how will ocean acidification affect coral reefs? And I'm sure you all know what ocean acidification entails with increasing CO2 in the atmosphere, and it's taken up by the ocean and it's making the ocean more acidic. The picture here, this is from a model projection of how seawater chemistry will change in the future under different CO2 emission scenarios. So approximately today, moving up to a condition where we have 650 parts per million of CO2 in the atmosphere. And the colors, they show how the saturation state with respect to aragonite is going to change. So if we're focusing on coral reef environments, sitting in this blue area of saturation state, we'll move into saturation state of this yellow area at the CO2 of 650, which is less than 3. However, an important aspect of these future projections of ocean acidification is that these model predictions, they typically apply to the open ocean. They do not model the actual conditions on the reef. And that is important because conditions on the reef are very, very, very different than conditions that we see in the open ocean. So what I'm showing here in this figure to the left here, this is the partial pressure of CO2 in the surface seawater at a range of different reef locations. So you can see this is from starting 2006 up until 2018. And what you can see is there's lots of variability over seasonal timescale, over daily timescale. The red line here, that shows the atmospheric CO2 concentration. So you can see for a lot of the time, many of these reef locations, they experience a PCO2 that's significantly higher. That's what you expect from equilibrium with the atmosphere. In the plot here to the right, this shows a whole bunch of carbon chemistry parameters. We have total alkalinity on the y-axis, PCO2 on the x-axis, and the color here shows the pH. typical open ocean conditions are shown by these solid black symbols and the variability of the open ocean. And the actual data points here, they show the conditions from a typical reef location. And as you can see, the reef experiences pH as low as 7.85 and sometimes as high as 8.25. The key point is that coral reefs in any nearshore environment, they experience radically different chemical conditions and variability compared to open ocean locations. So if we want to understand how ocean acidification will affect coral reefs, we first need to understand what the current conditions are and what are the drivers of that variability, and how are those drivers going to change in a future world that is warmer, higher CO2, and there's more acidic. So a lot of what we do in my lab is that we're going to different reef locations, or we're trying to work with collaborators in different countries to try to get as many different measurements from different reefs and different habitats. And the pictures here just shows some examples of reef locations around the world and different habitats where we have deployed sensors to monitor how, for example, pH and oxygen vary on temporal scale. But we also do big spatial scales, as shown here at the bottom, where we're really trying to see what is the variability across an entire reef. So not just in one place where you typically have most of the corals, even in the back reef and in the lagoon where you may have mostly sand. So this is something that we spend a lot of time doing. When we go to different reefs or when we get measurements, we also want to assess whether the reef, is it net growing? Is it making reef structure, or is it losing reef structure? And the way we estimate that is by chemical means. We measure this property called total alkalinity in the water, which we compare to offshore. And based on the difference, we can say whether a reef is net growing or if it is losing material. So we call it that we're taking the pulse of a coral reef, essentially. And what you see in this figure here, these are data from about 24, 25 different reef locations around the world, showing essentially the delta total alkalinity, the difference in alkalinity between the reef and the open ocean. And any blue data here means that the reef is calcifying, it's making reef structure. Any of the red bars here shows that there's actually dissolution, that the reef is breaking down and it's actually losing material. So as a result of ocean acidification, we think that these data are gradually going to switch up to this part here, to the red part, where reef will start to lose more material. So this is something we want to monitor and keep a track on, and look at how much do reefs grow and how much are they losing, and how is this changing over time. As you know, lots of reefs have been lost for various different reasons, and many scientists are now thinking about how can we stop this? How can we preserve our reefs? And there's an increasing push to actually do active interventions. And one of those interventions that have been suggested is, for example, seaweed farming, whether seaweed farms can benefit the coral reef. So that's really the project I'm going to talk about today, and can seaweed farming mitigate ocean acidification? And the underlying hypothesis for this comes from the fact that seaweeds, they are photosynthesizers, they take up a lot of CO2, and in doing so, they elevate the pH of the seawater. So we have our seaweed farm potentially creating favorable seawater chemistry for corals, here in potentially a coral farm, but it could be coral in the natural environment. And fishermen in Japan, they have done this for quite a while, and I know there's many seaweed farms in Indonesia as well. But in Japan, they often couple these seaweed farms with coral farms. The fishermen know that there's some benefits to both the corals and the seaweed farms by having them together. But they don't know exactly what that is or what like the chemical condition is. So that's one of the questions we want to address. And the pictures here are showing the seaweed farm and the seaweed called mozuko that they grow in Japan. And up in the left here is just an example of a table of from the coral farms. Regardless whether seaweed farms can be beneficial to corals in the context of ocean acidification, there are plenty of benefits of having seaweed farms and coral farms. The coral farms, for example, they can help to with the reef accretion, or it's a source of calcium carbonate, so it can help providing and building reef structure. It provides a habitat that can provide a habitat for fish, crustaceans, and other potential food sources. The farm corals can increase the diversity of sensitivity of the different corals available, so you can increase the resilience or the resistance, for example, of an environmental perturbations. They can also provide new larvae and rejuvenate an area that has been affected by a perturbation. And obviously, a happy reef is beneficial to human communities through tourism and fisheries. And a coral farm can then also create essentially job and financial revenue. The seaweed farm has similar benefits by creating financial revenue and job, and also providing healthy nutrition for humans, and is also part of blue carbon. So there are lots of benefits of seaweed and coral farms, and whether there can be a connection between the two in providing benefits from ocean acidification is then a question we're trying to address. So the overarching scientific questions of this project are: can seaweed farming and harvest effectively mitigate ocean acidification on coral reefs? Can coral farming produce sufficient calcium carbonate to sustain positive reef accretion? And can we predict ecologically relevant ocean acidification for any coral reef habitat based on reef properties and readily available measurements? So what are some of the scientific questions that we want to address, and how are we addressing them? So quantifiable questions are shown here on the left. For example, what is the mean and the variability of seawater CO2 chemistry across time and space at different reef locations? What are the physical and hydrodynamic properties of each reef locations? And what is the calcification dissolution potential of each reef? How close is a reef to the threshold of being net dissolving, or how much calcium carbonate is it actually producing? So to address these questions, we usually deploy a lot of physical and biogeochemical sensors in relevant habitats. We do drifter deployments where we may measure chemistry along the drifter path to look at the metabolic footprint of the reef. We may do reef scale surveys to assess at the ecosystem level what the calcification dissolution potential is. And we're also working on developing algorithms that will allow us to predict the ecologically relevant ocean acidification or chemical conditions at any reef locations. So for the next of my talk, I will give you some examples and show you examples of these measurements that we are making. So this is our study location in Okinawa in Japan. This is more or less a fringing reef with a back reef and a little bit of a lagoon behind it. And the area circled here by this green circle, that's the area where the fishermen have their seaweed farms. And these more elliptical circles, that's where they have the coral farms. So for our first approach in deploying physical and biogeochemical sensors, here are some examples of some sensors that measures pH, dissolved oxygen, temperature, and salinity, deployed in the coral farm in the top and the bottom, adjacent to the seaweed farms. And for the pictures to the left, these are what the sensors looked like when we put them out, and to the right, a year later, where they essentially become part of the reef because of biofouling. Here's some examples of some measurements with respect to temperature, salinity, light intensity, pH, dissolved oxygen, and tidal height. And if we just look here at pH and oxygen, the three different colors represent three different sensors, one at the reef flat, one in the lagoon, and one at the outflow channel. And as you can see, there are some differences as the water flows across the reef, crossing the seaweed farms, crossing the coral farms. There are modifications done to the chemistry. We don't know how much of that is due to the seaweed farms at this point, but this is looking at the reef at large, essentially. Then we also have a lot of physical sensors. The physics is critical. If we want to know how much the chemistry is changing and whether it can be buffered against ocean acidification, we need to know how long does it take for seawater to flow across the reef. So here's an example of an aqueduct that measures the currents in the water. And below here are some of the data where the arrows, the pink arrows, shows the predominant flow of the water, with the ellipse showing the variability. So across this area, you can see that the predominant flow during the time when we had these sensors is really along the reef this way. And you can even see that there's like darker patterns in the bentos that really reflects the predominant flow of the water. So even without instruments, we can, by looking at the bottom, we could say something about the flow in this area. And we also deploy these drifters. You can see in the picture here in this upper right, they're called reef drifters. And below, we have the lines following these drifters. And you can see how they're crossing the reef and coming out in this southern western part of the reef. When we deploy these drifters, we also make measurements of the chemistry to try to say something about the metabolism of the reef, whether it's net photosynthesizing, if it's net respiring, or whether it's net calcifying. And here's just some preliminary data where in the top panel, this shows the net community calcification, and the bottom shows the net community production. The data point I highlighted here represents the red and the orange drifters here, who happen to go right over the seaweed farms. And what we can see as over the seaweed farms, there's net production of organic matter, which we were hoping for. And there's also net calcification with pretty high numbers. So there's both net calcification and net organic carbon fixation going on as we made this measurement. But this was during daytime. And when we assess this, we need to think about both night and day, what's going on. Then finally here, this is showing some examples of the spatial surveys that we conduct, essentially looking at the difference between the open ocean and the offshore conditions, where the top here is showing changes in alkalinity. And as we move from offshore to inside the reef, we see essentially a drawdown of alkalinity during the daytime, which signifies net calcification. And the bottom here shows the drawdown in dissolved inorganic carbon, so essentially CO2, showing the autotrophy of the system, or the uptake of CO2 from photosynthesis. And the bottom here, we see dissolved oxygen, and we see an increase in oxygen as we move across the reef from photosynthesis. Another way to look at this is to make this plot I have here on the right, where we have total alkalinity on the y-axis and dissolved inorganic carbon on the x-axis, I should say TA on the y-axis. This plot is telling us whether a reef is net dissolving. Any data point on top of this red line here suggests that the reef, there's net dissolution going on at that time. And any points below shows that there's net calcification. And we can see our are data points along this line. Most of them take place under this red line, suggesting that there's net calcification going on over this reef. Anything to the right of this black line means there will be net respiration, and anything to the left suggests there's net organic carbon production. And because most of these samples fall on this side, during these sample events, it's net fixation of carbon essentially. But these were done during the day, so this is only one side of the story. To get a full picture, we also need to look at nighttime, what's going on. So the final thing that we're focusing on as part of this project is developing algorithms or models that would allow us to predict ecologically relevant ocean acidification. And why are we trying to do that? Well, first of all, it's prohibitively expensive to monitor and characterize chemistry of all reefs and habitats that are ecologically and economically relevant. It's just too expensive. Here are some of the different instruments that are used to monitor ocean acidification in coral reefs. The buoys, pH sensors, they all cost anything from $30,000 to $50,000 with enormous cost every year to maintain them, and only in one place. Similar, if we collect discrete seawater samples, we bring them back to the lab and we have these instruments where we can analyze the samples. Same thing, each sample costs about $100, $120 to analyze. That's a lot of money. We can't do that everywhere. So we're really trying to work on developing algorithms where we don't have to measure the chemistry everywhere. We can use some easy accessible parameters to try to approximate what the pH conditions are. So to describe to you how we do that, I'm first going to talk a little bit what are the controls of pH variability. So in these boxes, in the middle here, if this signifies the reef pH in a reef environment, this is first and foremost a function of what is the open ocean pH. What are the conditions in the open ocean? And as this water flows onto the reef, it's then becoming modified by different metabolic processes on the reef. So net community calcification or organic carbon production. So photosynthesis, respiration, calcification, dissolution. And that is a function of the community composition of the biomass and lots of physical properties. However, this is not the only thing. We also need to think about physical processes, including flow speed, residence time, the flow trajectory, the depth, and the morphology, because the time the metabolism have to modify the chemistry is going to determine how much the pH is going to change. On top of that, there may be downstream processes that modify the chemistry, and water may be entrained back into our reef of interest. And then we also have gas exchange. Gas exchange, though, is typically insignificant compared to the effect of all these other properties, so we can often ignore it. For these, the metabolic components, we've spent quite a lot of time trying to categorize that and look at how different communities modify the chemistry. So here's an example of an experiment we did in Hawaii in these type of mesocosms, where we put a whole bunch of different benthic communities in them, and we measured the rates of net community calcification and net community production to see how they modify the pH. And if we just focus on one example, for example, down here with NCP under current conditions, so it's not acidified conditions, we can see that the green line, which corresponds to fleshy algae, or the blue line, which is a mixed community, they had the highest NCP rates compared to any of the other communities, and therefore had the strongest modification of pH, essentially what we'd expect from a seaweed farm. And for all of these, you can see there are distinct differences from the different communities. The difficulty with this, though, is that once we go out into a natural environment, and here's some example from Hawaii, even if we make measurements in areas that have different benthic communities, shown here by these gray lines, and this is showing pH now. The colored lines show the results from the mesocosm experiment, and the plot to the left represents summer, the plot to the right represents winter. The problem when we make this measurement in the field, we cannot distinguish strong differences between different benthic communities in terms of their effect on pH, and which is perhaps very surprising. And it turns out there's other things that have a stronger control. The different communities still have a different effect, but there are other factors that are more important. And what those are has turned out to be depth and flow rates, for example. So here's an example of a study we did in several different reef environments where we deployed a number of different pH and temperature sensors to measure the short-term variability. When we looked at these data and we assessed them as a function of depth, so shown here to the left is the diel pH range, so the variability, and on the X is depth. You can see as we get to closer depths, so essentially depth less than 4 meters, a lot of data starts to converge to a similar variability, actually. And the same is true for temperature. So at shallow depth, the pH variability in any habitat on a reef can be relatively well predicted as function of depth. And this was surprising to us. And the reason, or that we think, is that wherever you go, no matter if you have corals, calcifying algae or anything, there's going to be primary producers capitalizing on the space, and they will do everything they can to photosynthesize as fast as they can. So for the short-term variability, these primary producers have a very strong effect on the chemical variability. So at shallow depth, what becomes important is the benthic biomass to the water volume ratio, essentially. And which is surprising, but it may be beneficial because based on this, we may be able to predict chemical variability by some very simple measurements. So instead of characterizing all these different things here, which is very difficult and very expensive, as a first approximation, we can reduce it to, if we know the open ocean pH and from physical process what the depth of the reef is, we can, to some degree of certainty, make a projection of what the reef pH might be. Given some of the resources that start coming aligned, for example, Ocean Data View and the global app database for different chemical parameters, we can retrieve an open ocean condition for many locations. There are also now techniques where we can start estimate bathymetry from essentially satellite imagery. So if these holds, we can now make estimates of the pH and future ocean acidification essentially for any reef habitat and any reef environment in the world by simple depth measurement. Of course, these are not perfect. There's some uncertainty associated with it, but it's, from our preliminary data, it looks quite promising. There are ways we can improve this, for example, by considering flow speed, depth, waves, wind, and tides, etc. As the time the water spends on the reef is really a critical factor of how long biology can modify the water chemistry. So here's just some examples. This is we're exploring right now, where we're looking at how the change in pH, shown here on the y-axis, how is changing as a function of changes in the flow speed, for example. So the flow speed is shown here on the x-axis. It's an inverse daily mean speed. So faster is over here and slower over here. So if there's slower flow, we see there's a greater modification of the pH. Faster flow, there's less time for the biology to modify. And the panel here shows nighttime. modifications, and to the right here shows daytime, where we also consider light, which is shown by the color scale right here. But then someone may think, but speed, how do you measure that? The current meters, they're also difficult to get hold of. And yes, I agree. But we can perhaps estimate significant wave height. So in this plot here on the top, you see the significant wave height measured outside of the reef, which could be estimated from a pressure sensor or from some rough models. And below, this is the actual measured speed on the reef, the flow speed on the reef. And by just looking at this, you can see there's a pretty strong correlation between the two of them. And by using this relationship, we have tried to predict, just based on the significant wave height, what the flow on the reef is. And the figures here are showing our predictions overlaid by the actual measurements, when you can see they're doing a pretty good job. So if we now can use significant wave height to predict the speed, we may be able to make our model even stronger to make predictions of how pH is going to change on any given reef environment. And I think this is critical as we're trying to make predictions for the future of how ocean acidification is going to affect reefs. We cannot rely on just the predictions for the open ocean. We need to consider what are the actual changes in the pH on the reef, which is going to be very different from the open ocean. So coming back to the overarching questions of our project here, can seaweed farming and harvest effectively mitigate ocean acidification on coral reefs? I haven't told you yes or no, and we are in the middle of investigating it, so I can't provide you an answer today. And the same is true with whether coral farming can produce sufficient calcium carbonate to sustain positive reef accretion. But I think I'm quite positive that we can fairly accurately predict ecologically relevant ocean acidification for any coral reef habitat based on reef properties that are readily available and that are not going to cost enormous amounts of money to make. So I think I'm out of time there. So I'm gonna stop my talk, and I'll be happy to take some questions. Yep. And I ended with showing some pictures of my collaborators in Japan here. While we tried to do some work during the COVID with some, mask requirements and other things in the field, which made it interesting. But I will stop my talk here, and I'm happy to take any questions.
All right, Dr. Andreas, thank you so much for your great talk. And I think it is interesting topic and regarding to the seaweed farming to mitigate the ocean acidification on corals. So we have so now we're gonna have a Q&A here. The participants or the people in the room, if you have any questions, yeah, you can directly raise your hand. All right. Is there any questions? Okay.
Don't be shy.
Yeah, don't be shy. And I have a question, Dr. Andersson. I think because you are saying that you were deploying a drifter, and then that you are measuring the calcification on corals, right? And but what my point is that when you are doing it in Japan, and then are you using like a specific coral species, or are you selecting the hard coral or soft coral for your research?
So the work we are doing is really looking at the ecosystem scale. So we're really interested in, let's see how this entire habitat is doing. Mm-hmm. A lot of work, is looking at specific species. Many people have taking the coral, look under different conditions and see how fast they
Yeah.
calcify. And that is very important work, right?
Yeah.
My lab, we are interested what happens at the larger scale. How does the whole reef do? And how do different parts of the reef contribute to the entire ecosystem? So if you look at this picture here, for example, there's a dark area in the upper right. Those turns out to be a higher density of corals. How does that area contribute to the entire ecosystem calcification and the modification of the chemistry? And then, of course, how do the seaweed farms down in the lower bottom and the coral farms contribute? But what is the total? So we are actually chemists. We are not biologists. So I actually don't care what species it is. Almost, I'm looking at the chemistry, and so I'm looking how is the chemistry changing, and by looking at the chemistry, I can tell you something about what's happening at the entire reef scale, and not only by what one species. Of course, that's also important, what species you have on the reef, because some species, they grow faster than others. Some species are more tolerant to heat stress, for example, or ocean acidification. And maybe, in the context of conservation, those are the species perhaps we really need to protect to maintain that there is reef building. But we are really interested in the large scale, at from the community to the ecosystem scale.
And yeah, I think it's just, because that I'm a biologist, right? And then so that I'm interested whether that is there any species impacted by the seaweeds. But the other question, I think, thank you so much. I think when you're saying it's like you look for the worldwide ecosystem when you done it in Tokyo, but do you try to observe the survival, growth or distribution, that I mean the specifically growth of the coral when there is any seaweed farming? It's close to the corals.
My lab have not done that, and we have only looked at seaweed farm. The first time we're looking at that is in Japan. So I would be very interested to come to Indonesia and work with you guys and see what's going on there. I remember I went on vacation in 2009 to Bali, and I saw some wonderful seaweed farms, and I remember my head was like, oh, I want to come and do science here, but I've never done it. So our other work has not been focused on seaweed farms. It's only been generally looking at how reefs, how they make their calcium carbonate, or how it dissolves, actually. Looking, because that's one of the predictions we have for the future, that dissolution is going to increase. So reefs are going to lose more and more calcium carbonate. And what does that mean for a reef? If they can't make the reef structure, that's a big problem.
Yes. A quick question, because that I'm still I'm still curious about this. And so I think it is a simple question, right? It is how maybe there is no good or bad, but how good is the seaweed farming to help the calcification in the corals?
I mean, that is the question, and we don't know the answer to that at this time. I would say so far we have seen elevated pH conditions, elevated aragonite saturation state conditions, right? Mm-hmm. Which we believe corals, they have high saturation states, they grow better. But even in a reef with natural primer producers, that seems to elevate it quite a lot, because the primer production is dependent on nutrients as well. And if you have too much seaweed, right, that's bad for a coral reef. Typically, so at least for this particular study, I can't tell you at this point, is it good or bad. The fishermen there, they certainly think there are mutual benefits between the two. But do I have a scientific, easy yes or no answer to that? The answer is no, not yet.
Not yet. All right, that I'm waiting for the next. I think we can invite you to here to see like what you mentioned previously. Okay, so there is another question from Muhammad Rifki Ramadan. So, and he said, you mentioned before that the ocean flow can determine the deacidification affected to coral reef. That's why the seaweed farming side by side of a coral reef is a good way to mitigate DOA. So his question is, does other factors like seagrass, habitat, or other things like breakwater structure can affect this too? And second question is, what makes the seaweed giving more better effect on preventing ocean acidification on reef?
So if I understood correctly, the first question was whether seagrass,
yes,
and also, yeah, in theory, right? If you have anything photosynthesizing, fixing carbon, that would drive up the pH. So any primer producer, whether it's seaweed, seagrass, some kind of macroalgae, just strictly speaking about the chemistry, during daytime, if you elevate pH, that would in theory be beneficial to the corals, right? At nighttime, there's a lot of respiration, right? And that could potentially negatively affect. So it depends on the balance, how much photosynthesis happens versus respiration. The beautiful thing with seaweed farms, right, is that it's being harvested. So we fix the carbon and then we harvest it. So you remove the carbon. So that's a blue carbon. There's a carbon capture there. And we could technically bury that carbon somewhere. So it's a permanent burial. If we eat the seaweed, we're putting it back up into the atmosphere, right? So there's no net change. But then that could, in theory, have potential benefits on elevated pH conditions on the reef. And I say in theory because it's, I think there are many other factors that we need to think about, and including how much do the corals think about or care about the pH in the seawater. We know that in the laboratory, if you take a coral, you put it in an aquarium, in a beaker, you lower the pH, they're going to grow slower. But if you do that in the field, if you change the pH where they have plenty of water flow, plenty of nutrients or food that they can grab and eat, are they as sensitive to the pH? And we don't have a really good answer to that.
Yeah.
The second question was whether there's a breakwater.
The second question is, what makes the seaweed giving more better effect on preventing ocean acidification on reef?
Yeah, so the benefit would be that essentially the photosynthesis, the primer production, that it takes up CO2, and taking up the CO2, it elevates the pH of the seawater. That's the hypothesis behind it, that it would benefit the corals.
All right. Thank you so much, Prof. Andersson. I think it's a great talk, and I hope that I can see you in Indonesia. And yeah, and I think that we finished this section. So Dr. Andersson, I think, do you mind that if you can stay, and then we have the appreciation before we are taking picture? Yep, operator, please. Can you stop share your screen? Yep, great. Yeah, we would like to thank you, Dr. Andersson, for your time to be a speaker in our program, Professor Discovery to Protect Our Ocean. So that we hope that we can see you in the next event. And also, please stay tuned, and then we're gonna take picture together. I think it's over the 300 participants here. So as our documentation. Yep.
Thank you so much.
Yeah. Yeah, we can. Yeah, we can take picture. Yeah, one, 2, 3. Okay. Next, we have many slides here, so stay tuned. Dr. Andersson. Yeah. Second, 1, 2, 3. All right. Next, 1, 2, 3. Okay. Next, 1, 2, 3. Next, 1, 2, 3. Thank you so much. And can we give applause to Dr. Andersson because his great talk. All right. Yeah.
Well, thank you so much for giving the opportunity to talk to you. And if anyone has any questions or anything, feel free to send me an email.
Yeah.
And if you want me to share my slides, I'm happy to do that too.
Absolutely. Thank you so much. I'm going to contact you later, Dr. Andersson. Thank you so much. Yeah, thank you so much for your time. I greatly appreciate that. Yep. If you want to stay in the Zoom meeting or if you want to leave, I think that's fine. Yep. And I'm going to, so we finished the second section from the second speaker. So I think now that I'm going to hand on to the MC. Yep. Yeah, MC. Yeah, please take away.
Okay, thank you, Mr. Tora, for facilitating our first and second lecture for today. And before we continue, here I have my partner who is going to accompany me as the Master of Ceremony.
Hello, sorry for being late. My name is Atma. All right, now we're gonna have some session of information that will be delivered by Dr. Yuniarti, our head department of Marine-FPIK University. But unfortunately, she is currently attending graduation ceremony, so this program will be delivered by Dr. Qurnia Wulansari. Okay, we're gonna hand it over to Dr. Qurnia. The time and screen is yours.
Okay, thank you, Miss Atna and Miss Regita as our MC today. Could you see my voice clear? Okay. Assalamualaikum warahmatullahi wabarakatuh. Good morning, all of participants who joined today's event. Thank you very much for this opportunity. It's really an honor for me to be here. I think we all are very blessed today because we can still meet virtually despite this situation to attend the PADOC or Professor Discovery to Protect Our Ocean webinar series that held by Department of Marine, Universitas Padjadjaran. I am Qurnia Wulansari. I am here as a representative, the head of undergraduate marine science study program. Today, she cannot join this event because he has another schedule in the same time. We apologize about that, but don't worry, because I'm here going to give a brief explanation about the undergraduate marine science study program. As we know, the undergraduate marine science study program under the Faculty of Fisheries and Marine Sciences, Universitas Padjadjaran. You can enjoy the explanation about the undergraduate marine science study program through the following video that we are going to watch after this. And recently in this year, we started to open international undergraduate marine science program. Maybe the operator can share screen the brochure. Okay, thank you for the operator. Universitas Pajajaran actually is offering 6 program in higher education through the IUP program or International Undergraduate Program. One of them is Marine Science Study Program. This program open for Indonesia citizen and international student to follow undergraduate degree in selected undergraduate program with uniquely designed learning method and to conform with international standard. For your information, the quota for international undergraduate program is around 10 until 20 of the total quota for undergraduate SMOP. SMOP is seleksi masuk Universitas Pajajaran. The undergraduate or IUP class, if you want to know more about this class in our university, you can check in our website WUNPAD SCID or in our faculty fpik.unpad.scid or in SMOP website through smop.unpad.scid. And to take a role in strengthening the marine and fisheries field in Indonesia and international recognition. Right now, we are heading the international accreditation, ASIN and IMARES. So we proudly to welcome you to our Faculty of Fisheries and Marine Science. Thank you for your attention and enjoy the video, and I return it to moderator or MC. Thank you. Play videonya.
The Marine Science Study Program at the Faculty of Fisheries and Marine Science, Universitas Pajajaran, is unique and emphasizes the fields of biotechnology, conservation, and hydro-oceanography studies. Biotechnology studies the science of utilizing marine life in bioprocesses, bioproducts, biomolecules, and genetics. Conservation studies the efforts to protect and maintain marine and coastal ecosystems, such as community structures and assessments of marine biota stocks. Hydro-oceanography studies science related to natural phenomena in the ocean, such as ocean waves, tides, currents, and the interaction between the ocean and the atmosphere on Earth. This is in accordance with the vision of the Marine Science Study Program of M.Sc, which has 3 research studies, including exploration of coastal marine resources or hydro-oceanographic studies, application of biotechnology in the utilization of marine living resources, conservation and coastal and marine resources, with a ESCO or Marine Bachelor's degree for graduates. The Marine Science Study Program received the superior predicate from Dept with SK number 2018/SK/BANTET/SK/PMT/S/3/2022 in 2022 and currently going on an international accreditation progress through IMARES and ASIN. From one of the international accreditation bodies in the marine sector, the Marine Science Study Program implements an independent campus curriculum development program in 3 main programs, namely curriculum development and restructuring, development and roadmaps, and technical guidelines for independent campuses, as well as the development of co-curricular or extracurricular syllabus and soft skills to support maximum lectures. So several laboratory facilities, namely a Marine Science and Technology Laboratory and an Integrated Biotechnology Laboratory. There are many activities carried out by the students of the Marine Science Study Program FPIK UNPAD in the field. For example, fieldwork practices in various agencies or conservation areas, and marine expeditions at home and abroad. Internships in Japan, as well as field trip activities to various areas, such as the Thousand Islands, Pumping Jawa Island, and many more. Job prospects that can be obtained from graduates of the Marine Science Study Program include being able to become government and private researchers, academics, consultants, conservation area managers, marine surveyors, diving instructors, NGOs, and CEOs of marine startups.
Mari adik-adik calon mahasiswa bergabung di program studi ilmu kelautan Fakultas Perikanan dan Ilmu Kelautan Universitas Padjadjaran. Ayo kita kelola sumber daya maritim Indonesia.
Thank you, Dr. Qurnia, for the wonderful information about the international class. Now we have our another lecture, which the lecture will be shared by Dr. Buntora Pasaribu, Ph.D. and Mr. Noir Purba, M.Sc. with our second moderator, Dr. Qurnia Wulansari. But before we start, we would like to read Dr. Qurnia's CV first.
All right. Our second moderator, Ms. Dr. Qurnia Wulansari. Her title is lecturer in Department of Marine Pajajjaran University, and her education is Environment Science Sriwijaya University in Palembang. Her expertise is marine biology, dynamics of the oceans, and marine life ecosystems. Okay, Ms. Qurnia is in our room right now. Okay, good morning, Ms. Dr. Qurnia. How are you? Okay. Hello, Dr. Qurnia. Good morning.
Okay, hello. Thank you, Ms. Atna and Regita, for introducing me. Before we started the session 2, maybe in here we have head of Department of Marine, Pak Sunarto. Can you hear me? Hello, Pak Sunarto.
Yeah.
Oh, okay. Thank you, Pak Sunarto, for your response. Maybe before we gonna start the session 2, you can give us some sentence for our event today. Thank you.
Okay, thank you, Ibu Qurnia. Assalamualaikum warahmatullahi wabarakatuh. Welcome to all participation. Thank you to Pak Buntora and Pak Noir as a speaker now, and thank you to all of committee in this event, and thank you to all participants. In this event, we will explore about all of our ocean resources, and we can hear all of the participants, all of the Yeah. And thank you to especially to Ibu Yuniarti and Ibu Qurnia. I think it's all. Thank you for all participants in this event. And I hope this event is not only in this year, and also we can conduct in the next year, in the next time, and we can also invite our presenter, other presenter from the other country maybe. Thank you, Pak Buntora. You can invite your friend from America and can speak here in our faculty. And I hope in the next time you can invite from the other country and we can give more information from the other country. Thank you for all participants, and thank you Bu Qurnia. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Pak Sunarto, for your speak. Okay, we will move to the session. Before that, Assalamualaikum warahmatullahi wabarakatuh. Good morning. Good morning, ladies and gentlemen, and also all of participants, students who join right now. Welcome to PADOC Seminar Session 2. First of all, I'm as moderator in the session. In addition, they will present about the theme future conservation, sustainable exploration of aquatic and marine resources. Right now, we have Pak Buntora. Pak Buntora, are you here? And Mr. Noir Purba. But before that, we gonna session with Pak Buntora first in the third speakers. Before that, I will read the speaker will be deliver the presentation around the 30 minutes. After that, followed by question and answer around 10 minutes. The participant can raise hand directly from the raise hand feature in the Zoom, and host or co-host will unmute your microphone. And you can also type your question in the chat box. You can use English or Bahasa, and operator will translate it for you. We will collect your question and answer in 3 incoming questions. And before we begin, I would like to introduce the speaker. He is Buntora Pasaribu. He right now as Assistant Professor Department of Marine Pajajaran University, and right now he is our lecturer, and his expertise in part of genetics and oil body in coral. And right now the theme of presentation is about the oil body. Hello, good morning, Pak Buntora. Please welcome Pak Buntora to start the presentation about the oil body biogenesis in marine plants. Pak Buntora, time is yours.
So much, Dr. Qurnia, for the great introduction, and also that I would like to say thank you so much to Dr. Sunarto, the head of our department, and for helping and also supporting this program, and also Bu Yuniarti and Bu Mega and all the staff member of a faculty of Fisheries and Marine Science, Universitas Pajajaran. So without further ado, that I'm gonna start my presentation. I'm gonna share my screen. Yep. I think, wait a minute. I don't know whether you can see it or not, but I can see my slide, but okay. But I can see it in the Finder. All right. So are you guys seeing my slide? Yep. All right. I'm gonna start because I only have a 30 minutes to present my research. So the title is the title of my presentation is "The Oil Bodies Biogenesis in Marine Plants," and so this is my outline. Is divided into the 5 sections. Is the first introduction, second aim, and third results. Fourth is summary, and fifth is the acknowledgement. First of all, that we know that we have been learning and that we have been using the TAG. What we call it is the Triassic glycerol. Is the main material of the lipid. But so far that we don't understand why the TAG is unstable and why the TAG is stable. So when the TAG's shape is stable is because they are surrounded by the proteins, what we call it the oil body proteins. Now that we want to know whether this is the oil body or not. So before we discuss about the oil body, that what is one of the basic things that we should know is the function of the lipids. So we know the lipids consist of TAG, and the main function of the lipid is the source of the energy, regulating and signaling, insulating and the protecting, and the most important thing is the storing energy. But one of the big question is, as a reserve of the energy, how are lipids stored and where are lipids found? So started from the elementary school until the senior high school, even after you are graduated from school, but you always use lipid. And then we do know that the lipids is a non-polar. What does it mean? And then that meaning is that do not dissolve in the water. But why? So if we look more detail in this slide, the structure of the lipid is mostly circle or micelle. But actually, the lipid structure is divided into the 3 parts, which is the amphipathic lipids, what we call it the single lipids, and the second is the lipid bilayer, what we call where we know that the hydrophobic and the hydrophilic they arranging themselves into the two layer, what we call it the lipid bilayer. So and the third and the last part of the lipid shape is what we call is the micelle. This is where that you can find this micelle when you are drop the oils into the water. But if we look more detail in the lipid structure, they are divided into the 2 parts. What is the hydrophilic is the head. What we call it in Bahasa is the kepala, means is a polar, which is the attracted to the water. And then there is a tail, what we call it ekor, is the resist to the water, right? And now that's the composition, the structure of TAG. So now what I want to show you is to answer my previous slide questions, how are lipids stored and where are the lipids found? So this is the picture from MRI scan, which is there is the 2 persons, which is one person is the overweight and the other person is a thinner. So like me, that I'm fat, so we can see that where the lipid stored. So mostly the lipid in human is stored in the lower body, is in the lower body part. It's in the hips, it's in your heart and also in your stomach and even in your shoulder. But if we compare to the thinner person, that you can see that the lipid is only accumulating in the hips. So and that's why we call it the less lipid. So the less lipid is shown in the yellow signal. You can see part in here. So and now how are the lipids stored? It is the lipid deposited as oil bodies in the cell. So the oil bodies accumulating, the circle of the oil body accumulating, and they will be accumulated in the greater amount in our body and make like a yellow signal in this picture. And then when you like to drink soybean milk, and then that you are also drinking the storage energy from the plant. So from this picture, what you drink every day, I mean, when you drink the soybean milk and then that you also drink the oil bodies, I think this is good for your health, right? And now that we want to say that oil body, how the oil body is stored in plants. So during the germination, in the early stage of the lipid, when the seeds start to grow, the seed will accumulate what we call it the TAG, and then the TAG will be degraded and converted into the glucose using what we call it the peroxisome, and then they will digest this lipid and use it as the energy. So next, when we say this is the picture of the oil bodies. So, and now the question is, what is the oil bodies? Oil bodies are the original composed of the mainly Triassic reservoir that you can see it from this picture, and surrounded by a monolayer phospholipids, you can see, and embedded with some proteins. And this is the stereoleucine A and B, caseoleucine, oleosin. This is what we call it the oil body protein, which is embed the oil bodies, make them compact and stable. And the other is that what the size of the oil bodies? Oil body is the nanomaterial. They are tiny. Their size is about the 0.5 to the 2.5 micrometers. And actually they are very stable because it's the consequence of the steric hydrons and electronegative repulsion, because it's being provided by the surface phospholipid and proteins. Now the question is that where we can find oil bodies. Oil bodies is mostly distributed, and you can find it in the seed, whether it's either angiosperm or the gymnosperm species, and also in the pollen, either is the angiosperm or the gymnosperm species, and also in the fern, and even in the primitive plants, what so-called algae, we can find the oil bodies over there. And so the oil body proteins has been found in the plants. So today the 3 oil body proteins is being identified, such as oleosin, caseoleucine, and stereoleucine, and they have a different molecular weight in their structure. First, oleosin is around the 17 to the 15 kilodalton, and the caseoleucine 27, and the newest one, oil body proteins found in the plant is so-called stereoleucine. Their molecular weight is about the 40 to the 39 to the 41 kilodalton. So this is the picture of the oil body protein structure, and we can see that the oleosin is what makes a difference in every oil body proteins is their hydrophobic domain, like I mentioned to you in my previous slide, which is that hydrophobic, it means like water, resist to water, sorry. So, and then the oleosin, they can anchor into the oil bodies because they have the 70 residues. And compared with the caseoleucine, they also have the hydrophobic domain, what we call it is the proline node. And this is, and caseoleucine can anchor into the surface of the protein. And so far, and then how the oil body proteins is targeting a mechanism. First, the TAG is accumulating in the smooth ER, which is the TAG is being synthesized, and also that the ribosomes start to translate oil body proteins, and they will meet in the end of the reticulum and the reticulum endoplasma. So, and they will bud, and then it is being and the bud, and then after that is being packaged, and then the TAG is being packaged and then embedded by the several proteins. So when then the oil body is mature, and they're gonna be released into the cytoplasm. So what is the application from the oil bodies? That we know that the oil bodies can be used to imitate the milk products, and also for the edible films, natural emulsion, and then that for the ice cream, and also even for the drug delivery. So the oil body has been observed in the pine seed. That you can see that the pine seed is one of the gymnosperm species that has been present in the dinosaurs era in the Triassic period. So that you can see when we observe the oil body under the transmission electron microscopy, you can see that the white circle, this is the oil bodies. And how we can isolate these oil bodies? This is the pine seed that we do the purification, and then we obtain the oil bodies on the surface of the water, and then the oil body is being observed under the microscopy. And we want to confirm that we want to observe the oil body, whether this oil body has the lipids or not, using what we call is the body P stain. And then you can see the green signal is the natural lipids. So, and the oleosin were also found during the duckweed hibernation. So, as we know that animal does the hibernation, but even the plant also did the same thing. So, in the duckweed, the oil bodies protein is being found. So-called the oleosin, there is a 5 oleosin is being identified in the aquatic plant. So, and also in the primitive plant, especially algae, that the oil body has been identified as well, which is related to the calysin. You can see that the oil body, the most of the chlorella intracellular cell is shown the yellow signal. This yellow signal is representing the oil bodies, and you can see that the oil bodies in the chlorella, I mean the green algae, they consist of the hydrophobic region or domain, and they also have the hydrophilic domain. But we can see there is the hydrophobic peak in this picture. But what that I wanted to tell you today is like from the primitive plant to the angiosperm, I mean to the highest and the modern plants, the oil bodies have been found. But the most question is, how about in the marine algae, seeds, or even in the marine flowering plants? Is there any oil body presence? So, like in the first speakers mentioned about the unbleached and the bleached coral, that you can see that the bleached coral sign the loss of the symbiont from the host tissue. Symbiont means the algae reside inside the coral's tentacles, and what the coral tentacle, what the symbiodinium or the algae doing in the coral tentacles. So actually the symbiodiniaceae live in the special symbiosome within the gastrodermal cells of the host. So it means the symbiodinion, the brown algae live in the gastrodermis layer, and then they will conduct the symbiosis mutualism to the coral as a host. So as we know that there is a close relation between the host and symbiodinion, and as we can see from this picture, the algae, the brown algae, so-called symbiodinion, is producing a photosynthesis product and send the photosynthesis product into the host and is being used by the host, and the host is provide the nutrient for the symbiodinion to survive. So there is the mutualism symbiosis between the host and symbiodinion. But our question is, this is the coral, right? Eupilia glabrescens, but our target is there any lipid droplets in the symbiodinion, and how we can identify them? So the first question is that we have to isolate the symbiodinion or the brown algae from the coral tentacles, because we know that the symbiodinion or the brown algae reside in gastrodermal layer, and we use the chemical, what we call it is the Perkel gradient, and then in the 80%, in the layer 80%, we will obtain the clean symbiodinion, and then we use the PCR and we design the primer to understand the clade or the species of the symbiodinion. And now we confirm that Eupilia glabrescens is a clade C symbiodinion. But our target is that we want to know, because when the symbion or the algae live in the free living, they lack of the oil bodies. But when the symbiodinion live inside the corals, we can see they produce more lipids. Another question is it the lipid droplets or not? So, and this is the comparison between the symbiodinian free living and the symbiotic, which is the lipids droplets can be stained by the Nile red. So, and now start from there, that we want to see the intracellular cells of the symbiodinium. We can see that the symbiodinion, the presence of the lipid droplets in their intracellular cells. So what we can conclude is the lipid droplet were distributed in the cytoplasm, which is closely related to the chloroplast in symbiodinion intracellular cells. If you look more detail, that the lipid droplet is close to the chloroplast and makes it difficult to isolate the lipid droplets. So the next is that we isolate, and then I isolate the lipid droplets, and then that we stain with the Nile red, and we can see that the presence of the lipid droplet were confirmed by the Nile red, which is stained by the natural lipids. And we try to detect the steric hydrons and electronegative repulsion, because we want to make sure whether the lipid droplet proteins presence in the oil bodies of the brown algae. So I mean, this is the picture of the oil bodies in the symbiodinion or the brown algae. So we can see from the video, means you can see there is the moving of there is a moving in between of the oil bodies. You can see this is because the steric hydrons and electronegative repulsion is caused by the oil body proteins on the surface of the oil bodies. So the next question is oil body present in marine flowering plants? So we know the seagrass, or Indonesian name is Volamun, is a unique flowering plants and highly productive submerged marine angiosperm that grow in shallow coastal habitats and play an important role. We know the seagrass played an important role in maintaining the stability of the marine ecosystem. But now the question, and now that I conducted the research in Taiwan, as the Dr. Andersson said, that we are working in the molecular biology, focus in the coral and also marine plants. So, and then that we took the sample in the Dongsa Island, that you know, the atoll I think most of the people know the atoll, and then it's like the ring. So, and then under this island, there is a seagrass bed distributed mostly. So this island is in between the China and the Taiwan. So it's like a dangerous island. So, and that I collected the seed, which is that we found the mostly seagrass species present is the Hemprichii, and then that I observe whether there is oil bodies present. We can see from this picture, it's just only a few oil bodies present, and then the purification is very difficult because mostly it's the carbohydrate. But we are successfully isolated the oil bodies from seagrass. And this is and then we try to align the structure of the oil bodies that we found. The oleocene is a presence in the and then the absence of the actin residues in the seagrass oleocene, and also that we draw the hydrophobic and then hydrophilic region, which is, we know that the hydrophobicity is anchored into the oil bodies. So what is the very unique about these oil bodies, oleocene in the seagrass? Because they also present the aprolanite motif that highly conserved among the oleocene. So I can summarize is that in the marine plants that we can observe the oil bodies, which is related to the chaliocene and also oleocenes, and also these oil bodies were observed as well in the brown algae or symbiodinion. So what we're gonna do in the future is that we're gonna do this for the future application. So thank you for your attention, and this is my acknowledgement. Thank you so much.
Okay.
Yeah.
Thank you for Pak Buntora with the great presentation about the oil body. All right, we are still waiting for your question in the chat box, or you directly can raise hand or unmute your microphone, please. Okay, Pak Sunarto, please.
Yeah. Thank you, Bu Qurnia, Buntora. It's interesting presentation, but I don't know about oil body deeply. I want to know what the difference of oil body from the sea, especially from the seaweed, and in the content and in the function, and we compare with source from the land plant, like bean, soybean, and so on. What the difference of the content and the function and impact of our body? Thank you, Pak Buntora.
Yes. So can I answer?
Yes, Pak Buntora.
Yes. All right. Thank you so much.
You can directly answer.
Yes. Thank you so much, Pak Sunarto. And the first question is there any difference the oil bodies from the sea and arable lands? So the first it is from the content, the oil body contents, there is no difference, because oil body composed of the TAG, protein, and phospholipid. But the most of the oil bodies composed of that three structure. And then but what makes it different between the oil bodies from the plant, from the sea, and also from the arable lands is their proteins, because most of the oil bodies is identified from the sea they lack of the proteins, such as they only have one proteins, what I can mention. So like in the algae, like dinoflagellate is identified from corals, is related to the calyocene. They don't have oleocene or even the sterilocene, but the oil body proteins identified from seagrass, they don't have calyocene, they only have the oleocene. So what makes it different? But in the arable lands, they only, we can identify the oil body proteins for the three proteins, oleocene, calyocene, and the sterilocene. But for the algae, they only have one or two oil body proteins. That's what makes a difference. For the future application, I think that we can use this the oleocene, one of the proteins from the oil body, even that we isolate from the ocean, from the marine plants, or even in the arable lands, that we can still use them as a food additive, and also that we can use them for the drug delivery, and also that we can use them especially in the pharmacy sector. All right, thank you so much, Pak Sunarto and Bu Qurnia.
Okay, thank you. Another question, maybe?
Buntora, so we can use the oil body from the sea is better than from the land plant. From the sea, lack of the protein.
But if the sea, the oil body from the sea is usually not stable compared with the oil bodies from the plant. So, but we can use the only oil body proteins, but we cannot use the for the whole oil body structure for the future application. That's a different, Dr. Sunarto. Yeah, Pak.
Yeah, thank you.
Yep. Thank you so much, Pak.
Okay, thank you. This is the interesting topic, right, about the oil body and the utilization for the pharmacy and also the health sector. Okay, another question. Maybe I have a question,
Yes.
Pak Buntora. Did you see the oil body composition after? I know you already researched about the oil body recent years, but did you different the composition of oil body in first your research and until now?
Yeah.
Okay.
That I can find it what I show you here is there is the different of the lipids when we found just like in the duckweed, like aquatic plant, they have less of the carbon in their lipids. So for the future biodiesel that you can use the duckweed oil bodies, but for the pine, you can see for the pine, that's why long time ago, I think that in China, they like 100 years ago, and then they eat the pine for the health because the pine consists of here. You can see there is a several that I show you here in the pine. The pine is now being sold in the market. Is really expensive in China and also in the US, because when I conducted the research, that's why I found why the Chinese people long time ago, and they always eat pine because there is a very good health for this, because they have the higher unsaturated fatty acid, which is the fraxinate acid, linoleic acid, adrenic acid. This is good for health. So that's why, and that's the difference. And the other is when I found in the for the algae, I don't show you in here, but from the symbiodinium, it's good for the biodiesel. So they have the different composition of of the TAG content in their lipid bodies. Yep.
Okay, thank you.
Yes.
That you say before, the oil body from the marine plants is not stable like the in the land.
Yeah.
How you can face the challenging and opportunity to get it?
Yes, I think it's pretty difficult because when I try to isolate the oil bodies from the plant, it's not stable, and it's when you isolate it, it's directly fused. And then, but what can I do is that I try to store it in the low temperature, so it make it the protein is more stable. So that's the one of the way that I can isolate the oil bodies from the marine plants. It is challenging for me, but to obtain the oil bodies from marine plants is necessary because we want to know the evolutionary the evolution of the oil bodies. Actually, in the plant in the marine is different with the arable lands. So now it makes clear for us, but it's a different evolution when the plants grows in the ocean and in the soil. Yep. Thank you so much, Dr. Qurnia.
Thank you so much for the brief explanation, Patora. And we have another question from Muhammad Rifki Ramadan. You said before that oil body in marine plants is more fragile. Correct me if I'm wrong. Will there be a negative effect if we use the oil body from the marine plant compared to land plant?
All right. Thank you so much, Rifki. I think it's not the fragile. It's unstable compared with the oil bodies isolated from arable land. But will be there negative effect if we use the oil body from marine plants compared to the land plants? No, it's not. So there is no negative effect. But if the oil body is unstable, it means they are quickly being used by the plants to conduct the metabolism and also growth. So that's why less oil body found in the marine plants or marine organism, because they're always moving compared with the oil bodies in the land, because they just only stay there and they are not moving. All right.
Okay, thank
All right,
you.
thank you, Rifki.
Thank you for the explanation, Pak Buntora. Maybe running out of the time, the question already answered by Pak Buntora. We move to the next agenda to give the certificate, right, to Pak Buntora as a speaker today, before we move to next presenter. And also, if you want to know more about the oil bodies, you can contact us in Kelautan at Unpad ACID, or you can contact directly Mr. Buntora. Okay, this is the certificate for Pak Buntora as a speaker in the PADOC seminar session. Okay. Okay, we will take a photo session first, Pak Buntora and this certificate. Please, operator, you can help me. Okay. Thank you, Pak Buntora. Okay, we are gonna to move to last speaker, but not least. He is also from Marine Department. He currently associate professor. You can share screen about the city. Okay, we have last speaker, Noir Purba, M.Sc, as Associate Professor, Department of Marine-FPIK. Now, he is Ph.D. candidate in University of Malaysia, Trengganu. Next slide, please. His expertise about the physical biology interaction, low-cost instrument, and marine debris and meteorology. Please welcome Pak Noir Purba, who will give presentation about the leveraging technology for marine conservation.
Okay, thank you, Bu Qurnia, for introducing me, and I really appreciate to Pak Sunarto as my head of department. So I'm trying to share my screen. Please, Bu Qurnia, if you can see my screen.
Yes, clear.
Okay, I will zoom it first. Okay, can I start now?
Yes.
Okay. Thank you, everyone. Good morning, because this is the last presenter. As Bu Qurnia mentioned before, my name is Noir Purba. My expertise in physical oceanography. And with me, there are many people supporting this research. Ibnu Faisal from Department of Marine Science, then Professor Widodo S. Pranowo from research, how to say, brain, research and innovation, I don't know, maybe brain. And last is Myron Kenzie from Ocean Institution. And my title is Leveraging Marine Technology for Ocean Conservations, and in the context of Indonesia as archipelagic country. So my presentation is like 28 slides, and I have 3 videos. So I don't know if it's fit for the today, but I wanna try to explain to all the participants because I know mostly of this participant is the potential students of marine science and also the undergraduate students. So I think this is also suitable for them to learn much about the technology, especially in the ocean context, and what we do and what will we do in for the ocean sustainability. So my slide also divided into 5 sections. The first one is the unknown ocean. The second is the biodiversity, because my title is related to technology and conservation. The third is protecting the ocean by technology. And the fourth one is marine technology. As I said before, that what we did and what we do in the marine science program. And the last thing is as the students, as the researcher, and also and the potential students, what you can do to the ocean by learning in the marine science program. So I think we all agree that the ocean is so dark. Before 1920, before World War I, I think before scientists or founder of the light, I think we still don't know about the what below the water. And in fact, our Earth consists of 70% of water and 30% of land. So until now, only 2% of the ocean seafloors have been comprehensively mapped. And recently, with technology such as like scuba divers, camera, underwater vehicles like submarine, satellite, and like also, we can see the beautiful of the ocean. I think when you snorkeling or diving in the island or in the beach, you can see the beautiful of fishes and also the beautiful of the coral reefs maybe in the 7 meters or 10 meters of the ocean. And from the technology, we can also see many creatures, especially in the deep sea. The snake is it has poisons, The second is the mola-mola you can see in Bali, and also many critters in the deep sea that we cannot see without light. So the technology is really help to explore the ocean, especially in the deep sea. And regarding to the context of Indonesian geographically, so we can see in the right pane, I think we have like coral distribution, mangrove distribution, and seagrass distribution, which is Indonesia has the high diversity in terms of the coastal ecosystem. Like in this morning, Andersson also say that Raja Ampat and all especially in the eastern of Indonesia, like Raja Ampat, Bunaken, Safu Island, and others island in Indonesia. So the fact also is Indonesia is home to 20% of the world fishes species and 15% of the coral species. This is our fact as Indonesian, like Indonesia as archipelagic country. But we have challenge to that ecosystem treatment. So the impacts from human activities is really high. And from previous literature, it mentioned that over 40% of the ocean surface is impacted from the human activities like marine debris, oil pollutions, anthropogenic pollution, and others. So one case in Bali, there are many tourists went to Bali and posting in many media social, and they said when they went to Bali, it's like holidays in hell, because there are many debris that they found in the beaches. So they have to clean it and until they lying in the beach. So the marine debris is come to the beach of Kuta, especially in December to February every year, especially in Asian monsoon. So this is also because debris is mostly from foam and also plastic. So I don't know the chemical reaction to the. I don't know how in details the chemical reaction to the ocean and also to ecosystem, but from the chemical expert that it can be harm to the ecosystem and also to the ocean. And the other things of our problem nowadays, we have many challenges. We have many problems in the global context, especially if we see the melting of the sea ice in the polar, the global warming, I think. And the second is marine debris. The third is like oil spills that harm biota and also mangrove. And the last thing is the ecosystem damage by human activities. So ecosystem damage is different from the ecosystem damage. For example, coral reef damage is different from coral bleaching, because coral bleaching is affected by the global warming, but coral damage affected by the human activities like diving and snorkeling or ships. Nowadays, in the context of Indonesian, also we can see that the Ocean Health Index, it's the Ocean Health Index is the comprehensive framework used to measure the ocean health from global to local scales. And the fact is, Indonesian is rank is like 137 out of 221 countries in the world. So it's really important to us to make the ocean of Indonesia to become a healthy ocean, the healthy oceans, by doing the monitoring, avoid threaten the ecosystem. And because my presentation is related to conservation, so I would like to show you the marine conservation area in Indonesia. We can see from Aceh and to Papua, we have many marine protected areas that this is protected by law from Ministry of Fisheries and Marine Science. And we can see the fact is the almost our marine protected areas is less protected or unknown. So the problem is there are many problems to this context. The first one is the located is too far from the mainland, and the second is we have not a good technology to monitor it. And the case to protect the ocean is by regular monitoring. I think as monitoring is the key to protect the ocean and to raise to enhance the ocean health. So in terms of the ocean conservation, the technological is needed and is the key for the monitoring. So as marine scientists or the oceanographer, what we can do for that? So our approach is to raise or to enhance or to develop many low-cost instrument, especially in terms of archipelagic country. If I don't mistaken by notice by Andersson this morning, that he also developed low-cost instrument, but it cannot be applied in the whole world. So he uses algorithm or he uses numerical analysis in terms of to support the instrument. So I think technology will help us to protect the ocean life, and also it help the human to synchronize to the ecosystem or to nature. So this is one of the technology that we use today. This is video. I will try to play it first. Is there any sound for this? Okay.
As we orbit the sun, Landsat 8 positions itself to capture imagery in direct sunlight. It's called a sun synchronous orbit. And it happens every 99 minutes and about 14 times a day. As Landsat travels from the North Pole to the south, it's capturing data over a 115-mile wide swath. So every day we add 14 swaths. And over 16 days, we complete a full picture of our planet. And we start the cycle again to watch global changes as they happen.
Okay, thank you. This is the first one. I think Bu Qurnia also expert in this one. And I hope Bu Qurnia will explain in the next session, in the next conference, seminar, I mean. And the second one is, this is a flip research vessel. And I think not many people know this. And I never saw this in previous presentation, so I think this is important to us to get information from this research vessel. So this is also video. I will show you a video. And this is the third innovation that like US and Australia and other European countries has developed gliders. The name of this instrument is gliders, and from what we heard from the media or news, that we found gliders also in Indonesia last time in Selayar Island, I think. So this is very comprehensive. This is very high-tech instrument that we use this instrument to measure many parameters in the water column. So I also make this in video, so we can see clearly what it's doing.
Submarine gliders make it possible to collect physiochemical data such as temperature, salinity, and oxygen levels from the sea in an autonomous and continuous way at a high spatial resolution. So SIB has a fleet of gliders taking samples continuously in the Mediterranean Sea with the objective of obtaining data, so its characteristics may be studied and small-scale variability solved. But do you know how a glider works? The glider is ready to start the mission. The sensors and the navigation and flotation mechanisms have been checked. Inflating the bladder allows the tail to rise up and receive the operator signal. The commands which initiate the mission can be received either locally from a vessel or remotely via satellite. The computer, located in the navigation bay, calculates the course to start the mission. At the same time, the scientific bay receives the configurations.
Sorry, Pak Noir, the sound is lost.
The piston which will permit immersion to begin. During the immersion, the glider is set to collect scientific data. The CTD allows depth and salinity measurements to be made. The fluorometer allows chlorophyll levels to be estimated. The oxygen sensor determines the concentrations of this molecule in the water column. All the data recorded by the sensors is stored on a hard drive. During the immersion, the navigation data is also registered. In shallow waters, the altimeter is used to avoid collisions with the seabed. When the seabed is detected or the depth objective is reached, the navigation bay gives the command to start the ascent. During the ascent, the glider continues to collect data measurements for temperature, salinity, and chlorophyll. The glider makes ascent and descent movements in the water column of between 0 and 1,000 meters on missions which can last up to 2 months, covering 24 kilometers a day in any meteorological conditions. In the case of an emergency or at the pilot's request, the glider emerges to communicate with the base. The bladder inflates once again to receive the operator signal. The glider transmits all the data collected, receives updates for the mission, and uses GPS satellite positioning to calculate its new trajectory before starting a new immersion.
Okay. There is another technology that used to track the biota in the ocean the name is biotagging or biologger. So it consists of GPS and also battery, and then it track the turtle or whale or other fishes in the world. So in the left side we can see the track of the turtle in the east coast of Africa to the, I think it's Egypt, right? Yeah, to the Egypt in the north side. So from this we can see where they go and where they come from. So it's really helpful to us to know the physiology, the habitat of the marine fishes. I think it's like 200 euros per instrument, I think. And like Andersson mentioned today, we also like logger, and in these pictures we can see the temperature logger and also the salinity logger that we can put everywhere, especially in the coastal system. So it can store the data into the memory card, and then we can receive it, synchronize it to our laptop. So we can monitor every time, every day, or every minute to monitor the ecosystem condition, especially to temperature increasing or salinity in terms of the climate change or global warming. So we can see here they use it in the eastern of Australia in Great Barrier Reef. So they know the stress to coral reef in their area. And last, in our program in Marine Science Department in University of Pajajaran, we also developed a low-cost instrument. We call it Drifter GPS Oceanography Coverage Area, or we call it REA. So the name of REA is also come from the Greek mythology, the daughter of the Gaia. So almost instrument that developed in the oceans by like US or European, they also name their instruments from the methodology Greek. So like Argos, Argoflot, I mean Argoflot, satellite, Jason and something. So we adopted the name from the Greek, and these instruments, the concept of this instrument come from the propagal of mangrove that floating in the surface every time, and then they will settle in the coastal area. So our instrument also floating in the surface of the ocean. So this is the picture of our instrument with the handsome guy in the left side. So I think the length of this instrument, I think, is 1 meter, and the weight is like 10 kilograms, and the sensor is in the upside. The sensors is in the up and below the housing, and it can measure the atmospheric parameter and also ocean parameter. The atmospheric parameters like atmospheric pressure, atmospheric humidity, atmospheric temperature, and in the ocean we measured temperature, salinity, turbidity, pH, and oxygen dissolve oxygen. And the battery can, I think it's like 3 months, can float in the oceans. And we transferred the data with Iridium satellite, and we retrieve the data into our portal, Pajajaran Oceanographic Data Portal. So you can also download the data freely. Later I will send you the link of the portal website. So the second instrument that we develop is Archaea. It means we add A in front of the Archaea. It means the advance, that the different Archaea between Rhea, that Archaea, the first one is it cannot be measure the atmospheric parameters because it will sink to the bottom of the water. So we can use it to measure many parameters. We can edit some of probes or sensors. We can see in the right side there are many sensors that we put in the top of the instrument, like pH, the black one I think is turbidity and also salinity probes. So we already tested in the Pulau Seribu, or which is located in the north of the Jakarta, and I also have the video for this one. So the depth of this water is like 10 to 20 meters. So it will sink first, and then go up with the blade. And we can see in the top is the sensors. It measures, like we can set it like 2 minutes or 3 minutes or half an hour. And then we have the depth sensor also, like gliders. So it can be measured the depth of the oceans. And then after when this instrument in the surface, it will send the data to the satellite, or with Iridium satellite, and then we will receive the data by email and then in the portal website. So also we collaborate this instrument with some of our partner in industries in Bandung, RoboMarine. So if you want to develop this instrument, or you want to adopt this instrument, you can discuss with us, or we can develop or add more sensors to this instrument. And this video is taken by Pak Ibnu Faisal, I think. His expertise in diving. He take this video from the bottom. The maximum depth of this instrument is like 200 meters. So it's different from gliders and Argo float. Argo float can dive into 2,000 meters, but our instrument is just like 200. From our discussion with many experts, especially RoboMarine Indonesia, but we don't have technology to make housing, to make the material that suitable to the depth pressure, I think. So we should collaborate with other countries, or we should buy the housing from other countries, I think, because this housing is made from Indonesia. So almost like 60% of this instrument is made from local content from Indonesia manufacturer. So this is the sketch of how to deploy the Archaea or Rhea. We can deploy it from plane and also from ship. and then it will sink to the bottom of the ocean, and then after that it will give the informations to us, and then it will go to the surface and then send to the satellite, and we retrieve the data from satellite Iridium, and then it is one cycle of this instrument, the process of this instrument, and then it floating when it is sending the data, and then after that it will go down again, and up to now it's like we have the battery lithium, and it can measure to like 3 months to 4 months, I think. So we have to. So this is also become our challenge, because the Argo float can can measure in the oceans like 7 years, and our instrument is just like 3 or 4 months. But if we compare to the in situ survey by research vessels, the research vessel when go to the sea, it will cost like 60 million per hour. So it is a low cost instrument compared to the if you go by research vessel to the ocean, especially in high seas, I mean. And the second is we also developed the Sea Ghost 2. The Sea Ghost 2, it is combined with the wooden, like how to say this, this is a floating also, this is a floating instrument also that we plant the GPS tracker like you use in the phone or in the vehicle, and then it will it will send the data, the position of this instrument. So this instrument is also simple, very simple. The purpose of this instrument is to track the types of many debris in the oceans, because as I said before, my expertise in the physical oceanography or applied oceanography, we try to track the pathways of the debris in the ocean. So because we cannot track the real debris, we make the artificial debris. So it will give us many information about the line, the path of the different debris. So for example, if you want to track the debris, the macro debris, I mean, we can add the weight in the bottom of the artificial debris, and then we can modify the weight of this artificial debris to make it similar to the real debris in the ocean. So it is, so we can mitigate or we can we can mitigate how to handle or how to tackle the debris, for example, in Kuta Island, so the tourists can come, and also for the long term, it is for ecological sustainability, I think. So I think this is my not last, but I think this is my end of my section. But what we learn in marine science, specifically, we do it for a better ocean. I think we can learn much from the ocean. We can learn technology nowadays, because many information, many sources that we can get. But the thing is that we can do it for a better ocean. For example, the momentum of this decade is the the ecosystem restorations in 2030, and the second is the SDGs 14. I think the SDGs 14 is life below water, and also SDGs 13 climate action. So the era of technology can be used to make the ocean better, especially if from my previous presentation we can see the rank of Indonesia is really low compared to other archipelagic countries. We have like 137 rank, and I think it's low. It's like football nation, I think. And other technologies that we can use, we can map the health of mangrove. We can also map the sediment transport or the sediment distributions in delta, in estuaries, and somewhere else. And with also we can see the many critters, especially in the deep sea, with camera underwater, with ROV. And also this is also important because many medicine that come into us is from the deep sea. And maybe we can also make the renewable energy from wind, from ocean currents, from tides and other sources. And also we can map the topography and artificial coral reef from the technology. So this is I think this is the last slide. I promise. So this is the future of ocean. So I think with the technology we can put, or we can deploy many technology in the ocean. But I hope that this instrument can give us many information about the ocean condition, atmospheric condition, and other sources to manage the ocean conservation, like how to say, coastal ecosystem like mangrove, coral reef, and also seagrass. So there are many challenges as oceanographer or marine scientist, especially in Indonesia, because I think we only have 150 universities, 150 program study in marine science, but we have faced many challenges, especially in Indonesia, because Indonesia is archipelagic country with 17,000 islands with high diversity. So it will be our challenge to make it better, especially in Indonesia. So thank you very much, Bu Qurnia. Handing over.
Okay, thank you very much for the great presentation. What a comprehensive presentation, Pak Noir, today. About the opportunity and challenging in our ocean to protect our ocean in the future with utilizing the modern technology. Okay, we are still waiting for the question. In chat box from the Muhammad Awaludin. Thank you for the presentation regarding the Indonesian government. Do they have the enthusiasm to implement this marine technology for the country? What are the recent updates policy in this issue, and about the area could you tell us in short the precision of or accuracy result based on the test? Thank you.
Should I answer now, Bu Qurnia?
Yes, you can direct answer this question.
This from Mas Yusuf, right?
From Pak Yusuf.
Yeah, okay. Thank you, Mas Yusuf. I think Mas Yusuf also know the answer of this question, I think. But because we have many participants here. The first one is do they have the enthusiasm to implement these marine technologies for I think for this country? Yeah, I have to support our government, Pak Yusuf, Mas Yusuf, because we also how to say, we also belong to the government, right? But I think we have to enhance our collaboration with the other countries like European and also to US. But I think from my experience, yeah, we need more effort to do that. So because they know that we have many challenges for this, but the effort is really like I said, we need more effort to do that. As I know now, we have we add 2 research vessels from, if I don't mistaken, from France that has in the Navy, Regal, and also BPPT also already have collaboration with Japan and other countries to monitor the tsunami, and I think we have to support them to increase the number of the technology to monitor the ocean. And the second question is the recent updates policies on the issue is like my presentation before is maybe you know about the gliders. We found it in the Selayar Island. I think it's 6 months before. I think that we are really because we have like political issue with China at the time. So the gliders measure temperature and salinity, but we know that temperature and salinity also can produce how to say the acoustic signal, right? So sorry, the sound of velocity. So the sound of velocity can measure from the temperature and salinity. So when you know the salinity and temperature, we can know where the depth of the where the suitable depth for the underwater submarine to through the Indonesia. So it's like the sailor zone in Indonesia. So it become the issue right now. And also we also how to say we also really how to say curiga to the other nations how to handle the data, because if they measure the water column from their instrument, we really curious how they get the data and how they process the data. This is the issues of Indonesia. So the government make really difficult. Sorry, how to say really difficult administration, I think, process, I think, so for that one. And then the last question about our hair, the precision of or accuracy result based of this test. I think in the lab test we already compare to the portable instrument, and the result is good, but not really precise, because we use the non-expensive sensors. But if you want to make it precise, maybe we can buy or embed the high precision sensors. But it will be need more money to develop it. I will show you later if you want the data from the probes, Mas Yusuf. And also, I think if you want to buy, I think because I'm also selling this instrument, I think this is 100 million, Pak Yusuf. I think you can buy it for us in the lab. Thank you.
Okay, thank you, Pak Noir. There is another question. Okay, Pak Buntora, you can directly.
Question, yeah, a quick question, because that I'm curious about that. how deep your archaea and rhea can go into the bottom of the sea, that they are still working? How deep?
Okay, rhea can dip. Rhea cannot dip. It's just floating in the surface ocean, but archaea can dip into 200 maximum, because the pressure is because we depends on the housing. Like I said before, that until now there are no industry in Indonesia can make the housing with depth like Argo float that can sink that can dip into 2000. So our instrument can dip into 200. But if we say say archipelagic countries like Java Seas or South China Seas, is just like 100 meters, so it can be used in the Java Seas and in the South China Seas. Thank you.
All right, thank you, Pak Noir.
Thank you, Pak Tora, for your question, and thank you, Pak Noir, for your answer. And there is another question. Maybe I have question, Pak Noir. Did you have experience get lost the instrument in the marine, and how to maintain? Because you said before the instrument will sink in around 3 months.
You mean archaea, right?
The data.
You mean archaea and rhea, right?
Yeah.
Yeah. This is also our concern. I don't know. Yeah, this is also answer to Mas Yusuf, because the instrument is really I think it's expensive for us, for developing countries, right? So we cannot guarantee it. We cannot guarantee it. I mean, I used to make it missing in terms of marketing. so like I said before, if we compare to if you want to go to the ocean by research vessel, you can compare it the cost. You have to buy our instrument like 100,000 rupiahs, and also you can go to the ocean by research vessels like 1 billion rupiah. So it will be more accurate. More effective using this instrument. So if you look at in details of our instrument, the archaea, there is a hook in the up of this hook of this instrument, so you can make it like you can be you can sorry, how to say, in order it not will be lost in the high seas, I think, in the ocean. So you can turn down by rope, and then it will be go up by itself. That's my answer, Bu Qurnia.
Okay, thank you, Pak Noir. There is another question? No? Okay, we reached the end of the session. The question already answered by Pak Noir. Because we are running out the time. If you still interested with this topic, you can directly contact noir.purba@unpad.ac.id. Okay. Before we end this session, maybe we have appreciation for Pak Noir, certificate for Pak Noir, and thank you for give us the presentation, and thank you for your time. We hope gonna see you in the next agenda. Okay, maybe I will comment the photo session with Pak Noir and the certificate. Okay. Operator. Okay, thank you, Pak Noir, and thank you for Pak Buntora. Finally, now we have reached the end of this presentation. I would like to say thank you for the presenter and for the great presentation and the participant, active participant, and thank you for your attention. And we have photo session with the participant and the speaker. The comment from MC, right? Okay. Please comment the photo session with the speaker, MC, Atna, and Regita.
No. All right. Thank you, Dr. Qurnia, for facilitating our last two speakers. Now we're gonna have some photo session. The comment will be on me. So for the first slide, 1, 2, 3. Okay, second slide. 1, 2, 3. Okay, that's enough maybe for the photo session. Once again, thank you very much, Dr. Qurnia, for facilitating our last two speakers.
Okay, thank you. I return it to MC.
Okay, so we now have Ms. Putri Utami as a representative of the Marine Conservation Master Program. Ms. Putri is going to present her research about community perception on mangrove ecosystem conservation in Labuhan Village. Ms. Putri, are you here?
Yes, I am here.
Okay, Ms. Putri, the screen and time is yours.
Thank you for your time. I will present about my research. Community perception on mangrove ecosystem conservation in Labuhan Village. I do my research in Lamongan Regency, East Java. Lamongan Regency has a total coastal area of 5,765 hectares with a percentage of the mangrove ecosystem area reaching 0.58% with a total area of 291.781 hectare. Labuhan Village is one of the villages that has the largest mangrove ecosystem area in Lamongan. This mangrove ecosystem in Labuhan has decreased in quantity and quality due to climate change in the form of sea level rise and increased human activities. The damage is exacerbated by the lack of community contribution to conservation due to lack of knowledge regarding the functions and benefits of mangrove ecosystems, and generally the assumption that coastal and marine resources are open and shared resources that are easy and acceptable exploited freely. Mangrove resources management is needed to prevent further damage. It cannot be implemented properly before knowing the perception of the surrounding community. Perception helps to make plans related to effective management, protecting natural resources, and improving the standard of living of people in coastal areas. So I gathered my data, public perception data, collected by purposive sampling technique. Data were obtained from respondents through a survey method with structured interviews guided by questionnaires. Respondents were 30 people, consisting of fishermen, fish cultivators, tourists, governments, and community groups. The result from this research is there are 14 mangrove species diversity in Labuhan, and there are 3 dominant mangrove species in Labuhan, that is Rhizophora stylosa, Rhizophora apiculata, and Rhizophora mucronata. So the mangrove ecosystem in Labuhan is fragmented by residential areas into 2 areas, namely Kutang Beach in the west, which borders the mangrove ecosystem of Lohgung Village, and Yaang Beach in the east, which border the mangrove ecosystem of Brengkok Village. Both are used as tourist attraction, but only Kutang Beach is managed optimally because access to Yaang Beach is hard and far from settled. So the total population of Labuhan Village is 7,682 people, with details of 3,523 males and 4,159 females, like villages located on the coast, the majority of Labuhan village residents works as fishermen. So this is the public perception. The first is mangrove ecosystem function. 30% they told me that mangrove ecosystem function for tourist attraction, 43.3% for erosion barrier, 10% for feeding ground, and 16.7% for storm protector. But the mangrove ecosystem utilization, 36.7% for fish pond dike and 63.3% for tourist attraction. Mangrove ecosystem management participation, 40% Labuhan people not involved to mangrove ecosystem management activity, 33.3% for rehabilitation, 10% for maintenance, and 60.7% is socialization. And they know cost of damage to the mangrove ecosystem is 33.3% cause abrasion, 10% cause deforestation, 40% for cause land conversion, and 60.7% cause erosion. And the conclusion for this research is community participation in mangrove conservation activities is quite good. However, efforts to increase participation community to manage mangrove in coastal areas must continue. Various factors that influence perception society is lack of knowledge related to ecology, low level of education, public awareness and time, lack of information on the importance of mangrove forest, and lack of education and socialization about mangroves. Therefore, hopefully, the government will raise an awareness community regarding the importance of mangrove forests through education and providing guidance regarding forest conservation. That's all my presentations. Thanks for your attention.
Thank you, Mrs. Putri. We've almost reached the end of our webinar today, but we have our Vice Dean for Researches and Organization, Dr. Mega Laksmini, Ph.D., here to give the closing statement for our webinar today. So, Dr. Mega, the screen and time is yours.
Okay, good morning, everyone. Assalamualaikum warahmatullahi wabarakatuh. So we are glad to see you all here today. We have learned a lot from the 4 amazing speakers today about their interesting work related to future conservation and sustainable exploitation of marine resources. Today's lecture is one of the approaches to strengthen the academic collaboration with the international institution to provide academic insight to students. I believe that during these lectures, all of us have obtained a very fruitful discussions. So on behalf of the faculty, we'd like to thank all the speakers for delivering such important knowledge and also interesting talk. Furthermore, we thank all the participants who have joined this lecture, junior high school, college students from UNPAD, and also researchers and also lecturers. We also thank all the committee members for organizing this event and ensuring that this event is proceeded so smoothly. So finally, we apologize for any inconvenience that you might experience during this event. Please visit our faculty website for more detailed information regarding the study program and also next year events. So thank again for joining us today. See you all next time and have a good day. Thank you.
Okay, thank you, Dr. Mega, for the closing speech. Before we end it, I think we are going to have a photo session with Dr. Mega. Operator? No.
Okay. The command will be on me. Put your biggest smile. We're gonna have some photo session in first slide, second slide, until the end of the slide. So, okay, I'm gonna count. 1, 2, 3. Okay, next slide. 1, 2, 3. For the third slide. 1, 2, 3. Okay, maybe that's all.
Okay, thank you so much for all participants who have attended PADOC 2022. Before we say our goodbyes, I'd like to remind you all to fill the certificate Google form that will be shared now by the operator team. I'm Regita and
Atma, as the MC of the event, representing the commit from the deepest of our heart, we do apologize if there are any mistakes that we made through the event. Once again, thank you very much for your kind attention. Please stay safe, stay healthy, and see you in another time.